Thank you very much for joining us. I know that you're all very busy people, but we do think this is a wonderful conversation that we're about to embark upon because a lot of the companies that are offering tools in the bioindustrial tech area do need to reach out to other smaller companies that need the capabilities that these tools are offering. So we are excited to be able to offer the platform where we can connect these companies together and build the ecosystem. But before we get started, I want to give you a brief overview on what we do at the ABPDU. ABPDU, which stands for the Advanced Biofuels and Bioproducts Process Development Unit, is a Department of Energy funded facility. We are funded through the Bioenergy Technologies Office. We're located in Emeryville, California. You can see this third floor here on the picture. That's where we exist. And as you may already know, Emeryville, California is home to many industrial biotech companies. So we are located right in the middle of it and help scale up technologies from very small companies, small scale companies, and work with anybody in the ecosystem, small and big companies. So as I mentioned, we are funded by Department of Energy through the Bioenergy Technologies Office to really understand and solve scale-up challenges. This is a fisheye view of our fermentation suite. We have deconstruction and a dozen or so downstream unit operations that I will touch upon very quickly. But we start at 250 ml scale with amber bioreactors, which is not in this picture. But we also have these 2-liter bioreactors and 300-liter bioreactors that is our maximum scale. As I mentioned, our goal is really to help novel technologies scale further so we understand and solve those scale-up challenges when a technology is being transferred from milliliters in bench scale to hundreds of liters in prototype level. Why do we exist? Most of the innovation that occurs in biotech right now occurs in milliliter scale or even lower, sometimes even at nanoliter scale. But the real world bioreactors with feedstocks that are cheap, that can help with the techno-economic analysis of each of these technologies, they actually look like this. The turbidity associated with the fermentation process itself really complicates how a culture performs in a bioreactor. So we have to re-optimize a lot of these lab scale technologies. And then let's not forget downstream unit operation. We have to optimize all of that as well. It's not enough to just produce the molecule. So we like to say we meet our collaborators where they are. Sometimes we work with people who only have done some shake flask work or milliliter level work. And sometimes we meet teams that have extensive process development experience and they want to operate at the 300-liter scale. We're happy to work with our collaborators where they are. And more recently, we've installed pressurized fermentation capability. I will not go into much depth here because you'll hear about this tomorrow in the equipment section of this expo. We have these HEL bioreactors that can be pressurized, and we've been able to show with Checker Spot and with Biomade funding that we can actually pressurize their technology and it works well even under such pressure. And as I mentioned, we also do upstream and downstream chemistry from milliliters to hundreds of liters scale, and we can go up to 10 atmospheres pressure. We have Hassel alloy reactors that can handle very high, low acid concentrations, alkaline concentration. We can operate under vacuum. So we can do some serious chemistry as well that goes along with the biology that fermentation offers. As I mentioned again, downstream processing is very important to us. We have a dozen or so downstream unit operations. The operations when we don't have an equipment and we need it for a particular company, for their particular technology, we rent that equipment. We rent it, we install it, we commission it, use it for the company, and then send it back to the vendor. So all the italicized equipment that you see here, they all belong to the rentals part of our operations. We also have some analytical capabilities, the basic ones that are needed to understand the processes that we are developing. When we don't have enough capabilities, we definitely leverage the other facilities at Berkeley Lab. If you're not familiar with Berkeley Lab, I would encourage you to look up the kind of capabilities that Molecular Foundry and Advanced Light Source offer. It's pretty cutting edge, extensively out there. So you can also engage with them directly by yourself. We are happy to make any introductions if you're interested. But when we need to use that one equipment to understand this one protein, whether it's structure or function, we can definitely leverage these capabilities that are out there. So if you were a customer, if you wanted to work with ABPDU, this is our initial contact. This is the first time you're hearing about us. We meet you, you meet us. It usually takes about 12 months to go from this, 12 weeks to go from the initial contact to the full contracting of a project and getting it initiated within the lab. And I must say that things change a little bit when we have foreign nationals involved in the company. But if it's an American company, US-based company with US nationals involved in the equity and in the management of the company, then it takes about 12 weeks. Excuse me. A part of the process includes developing detailed statement of work. We go down to resolution of 4 hours of labor needed to conduct a particular subtask. So our collaborators can really see what is it that they are going to pay for and pick and choose if they want to do something by themselves or they don't think it's necessary. And then from that point, from project scoping, we go to the formal budgeting process and contracting. There's extensive EHS review as well. We want to make sure that we are actually operating these novel technologies at scale in a safe approach. And what we have learned in the past few years is that our safety reviews have helped the companies understand their technologies very well as well, and then go on to operate safely much further beyond the ABPDU scale. Here's an example of a company we worked with. Hue was co-founded by Tammy Sue and Michelle Zhu. Tammy and Michelle came to us and said that they wanted indigo dye that they're developing through fermentation to showcase in a demo day at their accelerator program, IndieBio. From the point of talking to them, we were able to initiate the contract in 6 weeks. I must say this is an exceptional case. We can't promise 6 weeks for everyone, but we could prioritize if someone really needed to start very quickly. And then you can see here the indigo dye that was produced at the ABPDU and purified was sent to Tammy and Michelle to test at an industrial scale, on an industrial scale equipment. And then it wasn't a pair of jeans, but Michelle was able to show off this denim cotton that was dyed with indigo dye from the ABPDU. So we've helped this way. We've over 85 companies. You'll see we've worked with the small ones. We've also worked with the big companies like the Total and Shell of this world. We're primarily biofuels, biomass, and waste conversion focused. That's why you see more of our companies there. But as the industry has moved towards materials and chemicals, we've been working in that space and also the food space substantially. Health became very important since COVID, so we've been working with some health companies as well, a limited number, depending on our capabilities and our particular specific capabilities that they need. Ag companies as well. We also work with our academic collaborators to solve some industry-wide problems that no one company can or will want to solve themselves. So if you're thinking that you have a research type problem that you want to work on with us, we're happy to co-write the proposals and bring in the academic collaborators needed to solve those problems at scale. We also write support letters and sometimes even co-author proposals to SBIR grants, not just to Department of Energy, but also other agencies within the US. So you're welcome to reach out to us if you're interested in working with us on one of those SBIR grants. So we've helped put 17 products in the market. Some of them are listed here. And that's really our goal, is to maximize the opportunity for the companies by minimizing the time it takes to go from an innovation to producing that first prototype and helping them put the product in the market. We conduct industry listening days. We've conducted 2 of these. These do not occur on an annual basis like our virtual expo, because we do get extensive feedback from our industrial collaborators. And it takes a little while for us to work on that and get back to them for new feedback. For example, one of the capabilities that industrial collaborators suggested we invest in 2019 was gas fermentation. And we've been able to install and commission the pressurized fermentation capability I mentioned earlier, which also doubles up for our gas fermentation capability. And we are in the process of commissioning our 20-liter reactor still. So if you have feedback for us, please do share. We're happy to take the feedback and work on it. So let's come to the topic of the day, which is the virtual expos. We conducted one virtual expo last year, which was focused on the theme of scale-up facilities around the world. So capabilities and capacity around the world. How can we access it? How can you all access it if you needed it? It was a very successful event, excuse me, with many attendees across the world attending it, 29 countries represented, and 125 plus for-profit entities, 50 plus non-profit organizations and universities. And if you missed it and you still want to view it, we have all the, not all, but most of the presentations recorded and made available in our ABPDU YouTube channel. You just have to go to YouTube and search ABPDU and you'll find our videos. One thing I do want to mention is that this meeting is also being recorded for the same reason. And for that, so keep that in mind. Please keep that in mind and keep yourself muted unless you are invited by the moderator to unmute and speak. Only the speakers will be invited unless there is a real reason for it. So please keep that in mind that this meeting is also being recorded. So it can be made available on YouTube. So again, coming back to topic of today, which is the Virtual Expo 2025, Software Equipment and Expertise. This is going to be a 2-day event, unlike the previous version. And the first day, which is today, is focused on software, data, and AI. We have an amazing set of speakers lined up to speak to you about what's going on in the scale-up world in software, data, and AI. I really hope you could stick through the whole day and listen to them. I've been very excited to see their slides. I do want to note that we have to make one change in the agenda. We switched the speakers from morning session 1 to session 2. Ishan Srivatsava now will be speaking in session 2 at 2:30 p.m. So if you're planning on listening to Ishan, please do be around 2:30 p.m. PST, Pacific time. And similarly, if you were looking forward to hear Quinton, he'll be speaking at 11:15 a.m. PST. Please keep that in mind. And we will drop the link to the agenda in our chat right now. And it is updated on the website. This is for you if you were planning this yesterday. And then that's not it. Please do come back tomorrow. Tomorrow we have another long list of speakers who have incredible things to share. Please keep that in mind. And tomorrow we'll be starting early. Tomorrow we'll be starting at 7 a.m. Today we're starting at 9 a.m. PST. Tomorrow we'll start 2 hours earlier at 7 a.m. PST. We'll be starting with equipment and the speakers mostly coming from Europe. So we'll have the speakers present at a decent hour for them. And then we have an expertise session. Again, speakers starting with our European friends and then moving to our American colleagues. And then eventually we'll end with another equipment session for our mostly American colleagues. We'll be adjourning at a similar time as today, 3 p.m. PST, 3:15 tomorrow. But remember that we'll be starting at 7 a.m. PST tomorrow, not 9 a.m. like today, 7 a.m., 2 hours earlier. Okay, so with that, I'll just come quickly to some instructions and directions for this workshop, expo. First thing, as I mentioned a couple of times already, please stay muted unless you are asked by the moderator. This expo is being recorded, so it can be made available via YouTube. It won't be available immediately. It'll be a few months before we can make it online. So if you want information, please do stick around, but please do stay muted unless you're asked to unmute. If you have any technical problems or Zoom-related issues or questions or concerns, please do write to us. We are monitoring our email throughout the day, and we'll monitor it tomorrow as well. Please write to us at abpdu@lbl.gov, and we'll be able to respond immediately. And while you are in the sessions, and if you are listening to the speakers and you have questions, please feel free to drop those questions throughout the sessions in the chat box in Zoom. The moderator will ask the questions for you, but you're welcome to ask the questions throughout the session. So speakers are expecting that you will ask the question while they are speaking. You don't have to wait until the talk is over for you to ask the question. And we are also going to stop at each contact slide of the speaker for a whole minute. So you can take your screenshots or take an image from your phone or write down. So if you have any questions for the speaker that you want to ask after the talk or you want to reach out personally, feel free to do so. That's the whole point of this expo. So we will wait on each contact information slide for a whole minute. So with that, we'll wait. First of all, I need to thank the ABPDU team. It's an incredible group of engineers and scientists and facilities and admin folks who do some really cutting edge scale up work, which allows me to be able to hold events such as this. So thank you, ABPDU team, for the excellent work. And I will stop at our contact slide, and now we'll wait for a whole minute before I hand over to our moderator for the first session, James Garner. James, it's all yours, and I'm happy to take questions.
Thank you, Deepti. Can everybody hear me? Okay. Oh, we have some questions already. John, did you have a question? Or were you
No,
just...
I was just trying to say I can hear you good.
Okay. All right. Great. Thank you. Fantastic. All right. Give everybody an opportunity to make sure that they have our contact information before bringing Gavin Thomas to the floor. Again, I'll reiterate Deepti's message. Please do type in your questions as we go, and we will make sure that those questions are answered. If we can't get to the question, we will certainly make sure that the speaker has an opportunity to respond to that question after the fact.
Okay, well, good morning to you over the pond. So I'm Professor Gavin Thomas. I'm from Old York, University of York in the UK. And I want to quickly tell you this morning about a small enterprise that we're running out of the University of York called MORF. And I'll tell you a bit about how it came to be and how we think this could be useful as a tool for small biotech companies working in the industrial biotech sector. So MORF originated from a major piece of academic work that was going on in the UK. This was a project that I led. It was funded by the UK government, funder called the BBSRC. And this was a project called Detox, which is a collaboration across multiple universities with multiple biotech companies who are producing small molecules in microbial systems. The idea of the project really was to understand product toxicity. So it was a fundamental project to use complex multiomics to look at or grow microbes in production-like conditions, and then either through them producing a molecule, citramycin was a control molecule we used, which was non-toxic, and styrene was a molecule that we used, which was highly toxic, or adding the toxin from the outside if it was a feedstock inhibitor, then monitor under fermentation-like conditions how the cells respond to those stresses. So we wanted to use all the omics to try and understand this. And of course, we realized to do this, we would need some kind of tool to integrate all this information that was built on the genome sequence of the organism that we were using. So this was mainly done in E. coli. I'm an E. coli biologist, worked on the organism for over nearly 30 years now, goodness me. And this is an organism that we know very well. So this was the project. Joe Webb led quite a lot of the experimental work, and Vicky Springthorpe, who I'll mention, was the lead developer of our tool. So we developed a tool that would allow us to capture this information. And what I really wanted as the PI on this project was something that really there was zero barrier to use. So I didn't want to have this tool that required a download and kind of installation of anything. Certainly didn't require any command line. I wanted anybody who was at the bench to be able to very easily, quickly access this information. So it was essential that the whole platform could be delivered entirely through the web browser. And this is what we came up with. So this is the project that we developed. This is our styrene production page. You can go and look at this. There's a URL at the top. This is matching public and published data we did on this process. And you can see here that we have a number of different types of data that are stored in MORF. Everything is underpinned by the genome sequence of E. coli, by which we can map transcriptomics data, proteomics data. We have some metabolomics data in here too, And actually this even scales to metabolic pathway analysis and genome scale metabolic models. And just to show you some examples of that in a minute, we start obviously with the growth data. So we had a very simple tool just for visualizing the fermentation and growth of the organism in the fermenter, and a way to plot all the online and offline measurements we were doing of the process parameters through the fermentation. And then, for example, this is product production of styrene. And then, of course, we had our omics data, which we have very simple online tools to analyze, and we can easily look at genes which are up or down regulated. There's lots of user functionality you can play around with here to query the data. And all of these dots are fully clickable. They're all linking to genes in the gene and the genome that underpins this database. So you can go and query any of those particular genes. So you can look at genes that are up and down regulated, and you can go and look at the gene page for that within the database. So this was a really complete genome-based solution using the web browser to allow us, as I say, to integrate and analyze this complex multi-omic data. And as I mentioned to you, the start of that process was a way to visualize fermentation data. We were using in-house fermenters for this, and we wanted a way, again, to share that data easily with each other and query that data and be able to compare and compare legacy data with new data. So we went to, and actually Joyce Bennett, who's the CEO of our little enterprise, went to some meetings, and we spoke to some small biotech SMEs, and they said, well, MORF sounds really cool, but actually we haven't got any omics yet. That might be a bit down the line, but actually how about your front end for the displaying the fermentation data? So because actually we've got loads of fermentation data, and it's all in Excel sheets, and we can't really do anything with it, and it's taking us ages to analyze it all, and we can't compare historical fermentations very easily. So we thought, okay, well, let's work with them. We got some funding from IBioReact, and the High Virals Renewable Network in the UK, and we produced a project with them to actually develop further this fermentation front end. So a project page for this company would look something like this, where we have the ability, we store all of their historical fermentation data, and again, they can very easily analyze analyze and compare different features of their fermentation using our setup. And the nice thing about this is we have, when we designed this, we want it to be really easy. If you are looking at this data in the browser, we have an image editor. So basically you can play around with the labels, you can play around with the axes, you can do whatever you want, and you can save it as a JPEG and stick it directly in your PowerPoint or whatever you're using to prepare your presentation for your line manager or whatever. So again, really simple, really easy to use. And we work with a number of small biotech companies now where we can actually produce really bespoke solutions for them depending on the different platforms that they have. This was an example. This is kind of an example of our fermentation solution. This is kind of we have now interfaces where we can take data from a variety of different scales of fermentations. They're all imported into MORF, and then they can be visualized and compared easily so that users can share data internally, or again, ultimately, perhaps if data is published, there's a way to easily release that data through using MORF as the platform to deliver that. So just to show a few little examples, we worked with one company where they were using BioReact. These are very small scale fermenters that are really neat, where you can actually control quite a lot of process parameters. So we've developed a little platform that we'll take an output from the BioReact software. We can read it into MORF, and we can immediately visualize in our web-based browser the outputs of these. And of course, we can do this with many experiments, so the user can compare experiments over time. They can look to see how things have changed, and that allows them to make decisions more quickly about what they want to do with their bioprocess. This is with another larger scale fermenter where, again, there's more data types coming off it. But again, fundamentally, the idea is the same. We take those complex files that are produced by the fermentation software, and we can basically pass them into our very easily analyzable web-based tool and allow our users to more quickly reach conclusions and understand how their process is working and how that's working over time. So really, that's just a quick snapshot of what MORF is doing with its fermentation front end. Ultimately, that's only using a part of MORF. So in the sense, everything is underpinned by the genome of the organism that's being produced. So if you want to understand, for example, again, at some point, collect other types of data. And again, because we have the genome and because we have the metabolic pathway analysis built into that, and potentially for some organisms, a genome scale metabolic model, there is actually a way, if you're getting online and offline metabolite data, to actually integrate that using MORF to try and understand what that means. So if you're seeing changes in byproduct accumulation or something, we can actually try and understand that at the biological level, because as I say, MORF is underpinned by a genome-based representation of this system. And really, the kind of people that we're a small company. I say we're looking to partner with, ideally, well, not anybody who's interested, but our ideal people are small companies where we can work with them in a very personal way to develop this interface for them to really help them analyze their data more quickly. We have quite broad expertise in microbial, biochemistry, physiology, genetics, and we understand microbial systems very well. So we can also help small companies with their general bioinformatics solutions and help them understand what their data means and help them make their bioprocess better. Okay.
Excellent, Gavin. Thanks so much. All right. And I think with that, unless there is one quick question, again, if there are questions after the fact, please do feel free to add them to the chat. Okay. All right, so with that, I'd like to bring on Pascal Vonlanthen from Securecell.
Yeah, good morning, everybody.
Good morning.
I will share the screen. Please let me know when you see it.
Yep, it's on.
Perfect. Well, first, a big thank you to the ABPDU team for, organizing this event and giving us also the opportunity to present on scale-up software, equipment, and expertise. My name is Pascal Vonlanthen, and I'm a business development manager at Securecell. So the title of my presentation is Automating and Digitalizing Bioprocess Development, a vendor-agnostic approach. So over the next 10 to 15 minutes, I will demonstrate with some case studies how we think at Securecell bioprocessing automation and digitalization should or greatly influence a scale-up, and how we can support customers. And also we would like to highlight that we collaborate with other vendors to realize this quite challenging endeavor. So maybe if you have never heard of Securecell before, here is a very quick introduction to the company. So Securecell was incorporated in 1995 as a spin-off from the Swiss Federal Institute of Technology. And over the past 29 years already, we have provided advanced bioprocessing, automation, and digitalization solutions to the bioprocessing industry. Securecell's headquarters is in Zurich, Switzerland, so that's where I'm based. And we also have subsidiaries in New Jersey, in the USA, in Leiden, the Netherlands, and in Lisboa, in Portugal. So let me start with this slide. So I think we can all agree that for an efficient process scale-up, we need a high degree of automation and digitalization in process development to increase the amount of meaningful data we generate and also improve then therefore process understanding. So my question is, or was at BPI West, what is the current status of the automation and digitalization initiatives of the bioprocessing industry? So on the left side, you can see charts showing the results of a survey from BPI West, where we ask exactly these questions to attendees, where they think at what stage their company is in terms of a strategy for bioprocessing, automation, and digitalization. So interestingly, 24% of the survey participants last year claimed that their company has a clear strategy for bioprocessing, automation, and digitalization in place, while over 53% claim that the companies are developing one or are considering to do so. So this indicates, of course, that bioprocessing industry is making good progress and heading in the right direction, but it hasn't reached its ultimate goals yet. And despite all these ongoing automation and digitalization initiatives, I think you can appreciate, especially if you work in the lab, that there are still a lot of missing interfaces in today's bioprocesses that prevent the implementation of automated monitoring and control tasks. So displayed here, I have a very standard workflow that I often see when I visit companies, which is comprised of manual sampling of the bioreactor, therefore manual sample processing, manual sample transfer to an at or offline analyzer, and thereafter often paper-based or Excel-based data management. With this slide, I would like to present how the manual workflow on the slide before can be easily automated. So if you add an automated sampling system that's here schematically displayed, all the manual sample processing, sample transfer tasks, and further manual work can be automated. If you also add, besides an automated sampling system, an overarching process information management system that connects and integrates all these devices, the analytical data can be automatically imported, assigned to the respective process in real time, and also centrally stored in a database. So this process data can be further used in automated feedback control loops for advanced data analysis. As you can see, compared to the slide before, we have here with just two tools, or one hardware and one software solution, a complete automation of a workflow. Of course, a short disclaimer here, both the automated sampling system Numera and the process information management system are developed and supplied by Securecell. So maybe that's a bit a Securecell-centric view, but nonetheless, I hope very informative for the audience. And I understand that today's focus is on scale-up software, and the equipment and expertise part follows tomorrow. However, we believe that, for an efficient bioprocessing automation, digitalization, and hence also a seamless scale-up, the hardware and the software need to work together in perfect harmony. And on this slide, I would like to quickly introduce you to the functionalities of the Numera automated sampling system, because it's mentioned in the application examples that I will show later, and that you know what it does. So the system can interface with 16 bioreactors in parallel. It aseptically draws samples, it dilutes and filters them, and it either stores them on a fraction collector or directly sends them to an integrated analyzer, which can be a cell analyzer, a biochemical analyzer, or an HPLC system. Also here we work closely together with various analyzer companies. Then, 2 slides before, on this workflow slide, I showed you that there is also a need for an overarching process information management system. And our software Lucullus is an example of such a comprehensive software that has simple SCADA functionalities, but also additional unique features. So just to very briefly also introduce it, at the core of Lucullus is its monitoring and control functionality. So currently we have integrated about 100 different devices from different vendors into the software. So this is really the monitoring, control, and SCADA part. Lucullus can grab the data from these devices, store it in a database, and also use it for automated feedback control loops. Then besides this monitoring and control tasks, Lucullus also supports a sample management functionality and a raw material management functionality. I will not go into detail here, but yeah, if you're interested, reach out. And what I want to highlight is especially this layer of data exchange. So I understand that a lot of companies already have some sort of software solutions in place, be it a LIMS system or maybe an advanced data analytical software. So Lucullus, of course, provides the necessary interfaces to exchange that data. And that's also what I mentioned at the beginning. I think collaboration here between the vendors is very important. With this slide, I quickly want to summarize my introduction and before going to the case studies. So what are the most important prerequisites and functionalities in our view a vendor should have to provide seamless automation, digitalization, and also help with a seamless scale-up. So we think a vendor should be to some point agnostic to prevent the vendor lock-in. The solutions need to be overarching, right? Connecting multiple equipments and also providing kind of a layer above the hardware devices for the software specific now. And then they should have open and standardized interfaces. I already mentioned that before. They should, of course, be scalable, growing together with the customer, and comprehensive, supporting end-to-end workflows. So here an example, I think a lot of software solutions just support monitoring, data gathering and then storing. We think that that's not enough. A perfect solution should also enable feedback control loops to have actionability on your process. And then eventually also, of course, the solutions should be secure to ensure data integrity. So before we continue, just a very short excursus. I think we have seen that Securecell is one of a few companies that can offer both a comprehensive hardware and software solution for bioprocessing scale-up. So in the slides before, I presented a potential solution with Lucullus and Numera to strongly increase the degree of automation of existing environments. So if there are already bioreactors around, if the customer has analytical equipment, we basically glue them together and establish a workflow. Now, what you see on this picture is Securecell's new end-to-end bioprocessing development platform called the Integra One. And you can think of it like an Ember 250 from Sartorius, but just with more functionality. So the platform features 32 to 64 fully automated small-scale bioreactors with a working volume of about 20 to 200 milliliters. The bioreactors are organized in so-called single-use reactor array modules that you can see on the top right here. And they are basically located in these upstream processing units. So the USP units can be, of course, automatically and aseptically sampled, and the sample transferred into a vial that sits on a conveyor belt that you can see behind the USP units. From the conveyor belt, the sample vials are then transported to a high-density filtration and sample preparation station where cells are removed, and also further to a PAT island. Here, this PAT island that can be populated with the analyzers of choice. Again, I highlight our collaborative nature to basically configure analyzers ranging from an HPLC system, cell analyzer, or biochemical analyzers. Now, we are, of course, aware that many customers are heavily invested into the Ember platform. So if you already have Embers, we can also provide this whole automation that you see by removing these upstream process units and just placing the Ember there and providing an interface to the sample bus. So for time reasons, if you're interested in learning more, please reach out. Now let me switch to the application example. So with the following first one, I would like to present how Emiris digitalized their complete bioprocessing development ecosystem. So what this slide should show is, that Emiris integrated all their bioreactors, which are from different vendors, including any peripherals, into the software Lucullus. So this includes small and medium scale bioreactors as well as larger scale pilot bioreactors. And now the question is, what is the advantage of such a setup? So again, all your bioprocessing data ends up in one software that can not only collect and harmonize, but also store the data and use it for advanced feedback control loops. With the second application examples, I would like to very briefly also present another customer installation, which is the Helmholtz Center for Infection Research in Germany, that uses Lucullus to establish its scale-up processes. So the Helmholtz Center for Infection Research is investigating secondary metabolites, so-called biologically active natural products. And the researchers are harnessing the biosynthetic talent of microorganisms to produce those metabolites. And one goal is to derive potential antimicrobic agent candidates. So on the top right, you can see again the network architecture. So the bioprocessing laboratory combines Lucullus with several integrated modern multiple scale fermenters, including shake flasks, Ramos shake flasks, laboratory scale fermenters, which are Eppendorf Dasket bioreactors, small-scale pre-fermenters from BBI Biotech, and production-scale bioreactors from Friends. And all this equipment you see here is again, of course, connected over the local network to Lucullus. And one of the most valuable functions for these researchers is that all the data, including data from connected peripheral devices, are securely stored in real time in a central database, ensuring full data integrity and complete documentation. This then allows the researchers to compare the data of running processes with historic processes to basically evaluate the process performance during scale-up. And this is important as some of these organisms have never been cultivated before, right? So you can use, data of a process that you ran 3 years ago, which has maybe a similar microorganism, to the one you are just testing now. So this is very helpful for them. And yeah, I see when I look at the time, I will just very briefly also mention the third application example, which is the closed loop control at Teva. So Teva is a generics company, and as you know, they have to be competitive, and therefore they rely on a high degree of automation and digitalization to enable a quicker scale-up of their bioprocesses. So what you can see here again is their network architecture of their laboratory environment. So they use, for example, the Numera to sample from bioreactors and transfer it directly to analyzers. All this data that's generated, from the bioreactors, inline sensors, and also these online analyzers ends up in Lucullus, where they have automation recipes running. And besides that, they also use the Lucullus Media Kitchen, where they register raw materials, and they manage to identify, for example, that if they change material lots, some critical quality attributes of their antibody changed. So it's really important to keep track of your whole process information. And yeah, this is one example how you could do this. And yeah, that's it. I will jump over the last slide. Thank you for your attention. I hope to see you at SynBio Beta next year, and please reach out if any questions.
Okay, thank you, Pascal.
Thank you.
And I'll just reiterate, please do put any questions in the chat. But with that, let's move to Ganymede Bio and our speaker, Gavin Smith.
Okay. Well, thanks everyone for joining today. We're excited to show you our platform, and Ganymede is a scientific data cloud. So we do a lot of integrations with instruments and different applications to bring data into one single data source. We've done some work with Berkeley Lab already, but are hoping to do more in the future. And the reason why we exist is that we see that before people bring on Ganymede, there's a lot of what we call human middleware going on in the lab, whether it's moving files via a USB drive, doing a lot of repetitive analysis in Excel or Prism, kind of those more legacy desktop-based softwares, and then ultimately moving data into an ELN or LIMS manually. And that presents a huge problem of being able to quickly and efficiently move data, make sure that your analysis is certified to be completely error-free. And then lastly, making sure that data is getting into an informatics system in a timely manner. So at its core, Ganymede is doing two main things: capturing data from instruments, from analysis apps, from CROs or CDMOs. We also do work with CROs and CDMOs to make sure that we're being able to serve customers' data very easily as well. And then running lab as code, which is right here, our paradigm to be able to analyze that data. So these are our data pipelines that are user-defined code hosted in our platform. And I'll be talking a little bit more about that in subsequent slides, but really that is the core of our paradigm, is being able to provide out-of-the-box connectors to different instruments. And we work with anything from automation equipment to analytical equipment, and we do a lot of work specifically in the bioprocess area. And then ultimately connecting to informatics systems that have out-of-the-box connectors as well. So what that actually looks like in practice is that we are typically connecting to an instrument via what we call an agent. We can also develop API connections via our API to analysis tools. We can set up connections to collaborators like CROs or CDMOs, and then aggregate all of that data, whether it's via OPC or via file-based connection, into our file system, where we can create a scientific data management system out of all that software. So once we're actually bringing that data in, we are parsing that data out into a tabularized format. So we believe in making data fair, so findable, accessible, interoperable, and reusable. The way that we do that is by parsing that data into a predictable format, standardizing the way that we're capturing that data so you can easily query it, and then being able to query it via SQL via data mesh. Once that data is actually parsed out in our platform, you can run what I was mentioning, our lab is code paradigm, to quantify and analyze data via Python scripts that you can develop and host in our cloud. So we offer multiple different versions of our platform. One of them would be to have Ganymede do everything as professional services. We have a team of really strong scientific software engineers that understand use cases quite deeply and are able to create custom analyses and parsing scripts to bring data into our cloud as soon as possible. Another way is to use Ganymede as a developer platform for folks that have more technical-minded people on their team to be able to use Ganymede to create some of these connections themselves, while still utilizing a lot of our out-of-the-box infrastructure and connectors to, again, either OPC or bio-based connections to different instruments. So once we have actually ran our flows in lab as code, what can we do with that data? We're automatically inputting data into something like an ELN. We're generating downstream files that may be used in an experiment down further in a workflow. We also have the ability to virtualize different apps in our platform as well. So this is a pretty unique part of our platform, where we're able to take traditionally desktop-based softwares and be able to virtualize them inside the Ganymede cloud to be able to then have a lot of metadata created when you currently were not able to see that before. So a big example of that would be something like FlowJo for flow cytometry. We could do that for different bioprocess softwares as well. But having that traditionally desktop-based app be hosted in our cloud would allow a seamless experience for a single window to be open in a Chrome browser or any web-based browser, and then be able to then do your analysis with a human-in-the-loop step after our flow has picked up the data and parse that out into our data lake. So if there is a need for kind of one human-in-the-loop step, we offer the ability to have those apps virtualized. When the data is fully analyzed in that virtualized app, when you press save, the data goes back into our scientific data management system. We can run more code on it and then have that go into our destination of choice. One other thing is that we sync a lot with folks' lab infrastructure and data infrastructure, although we do provide, as I was mentioning, this data lake. And so people rely on Ganymede for a lot of their data storage. We kind of live in a middle ground of hyper specialization and more front-end facing app like an ELN or LIMS such as Benchling or LabVantage, or these more industry agnostic cloud providers such as Azure, Databricks, AWS, what have you, because we have a lot of these scientifically geared out-of-the-box tools, but also still the flexibility to run user-defined code in our platform. So we view data maturity as kind of this pyramid here. So we start with creating a system of record. A lot of these for folks are already in place software such as an ELN or LIMS or an MES, and building a data capture system on that with Ganymede's SDMS product is kind of step one to building a more robust data strategy. As you bring on more and more of your instruments and start bringing on more data capture into your data strategy, then you can start to create these data pipelines, more automations, And then ultimately, on top of that, be able to run managed AI in our platform as well, build a digital twin of your lab, and then be able to have kind of by the second views of data that's being ingested into your lab as well. This is a case study of one of our clients, Chiappetta, who were doing a lot of that kind of human middleware steps that I mentioned in the beginning of the talk, where people were moving data from flow cytometers, cell sorters, this could very easily be bioreactors, and putting that data into things like Excel, Google Sheets, what have you. A lot of that data was unstructured, it was unpredictable, and it took a lot of time to curate that data, to pick out time points, and then ultimately get that into their electronic lab notebook. From there, they were bringing that data back out of their electronic lab notebook and then putting it back into Excel to make a compiled QC data set. This process overall, at the top of the slide here, would take on the scale of many hours. After employing Ganymede, we were able to shrink that down to just a couple minutes, right? Of the second that the data was created, we were bringing that into our cloud. We were analyzing that data immediately. We were entering it into their ELN, and then pulling that data from the ELN to compile a QC data set automatically. This slide is showing a little bit more of kind of the architecture of what LabasCode actually is doing once we're acquiring data. So I mentioned agents before. These are a pretty common way for us to connect to instruments. Again, we can connect via an API or OPC as well. But really what these agents are doing is watching for any data that's coming off of the instrument and is sitting basically on the instrument itself or a PC that is connected to the instrument for any data that's coming into our system. From there, we're bringing that data into our scientific data management system, where we can contextualize all of the files and data with tags. So we can apply customized tagging via those agents to say, okay, what instrument did this data originate from? Who did it? At what time? Is it does it have an experiment ID in a different data source? We can create hyperlinks and really nice contextualized kind of matrices to make sure that you're able to find that data very easily. Once that data is in our file capture system, we parse that out with our LabasCode paradigm to be able to tabularize that data into our data lake. From there, that allows you to create visualizations on top of it. So we have a really nice dashboarding tool as well, but also to run more robust analysis via those Python Jupyter notebooks, and then ultimately get that data into an ELN or LIMS. From there, we can actually use the inputs into an informatics system to trigger downstream flows, to kick off a new run, to send run instructions, to send work lists, things like that, to downstream instruments, all while keeping all of that data saved forever in our data lake, as well as syncing to your current data infrastructure. We can also make a lot of these powerful dashboards, not only for the raw data that's coming off of instruments, so you'd be able to see that data coming in live via our dashboards here, but also be able to monitor all of that data with the kind of asset utilization that's going on with any runs that you're creating. So we can monitor things like the number of runs that were done that week, number of users, are instruments online or offline. If you have multiple sites, we can monitor the amount of runs or instruments that are being used per site, and then ultimately deliver these real-time dashboards to you as a data product. So scientific data clouds are becoming standard, and people are adopting softwares like Ganymede, ultimately for kind of three main reasons that we see. One, we're saving scientists a ton of time. So for this 20%, honestly, I think is pretty conservative. We have reduced workflows from people that would take multiple days or like 12 hours into the course of 2 minutes. So ultimately, we're removing a lot of repetitive tasks and letting scientists focus on science instead of wrangling data and doing repetitive tasks in things like Excel. We're also ensuring data integrity. So making sure that we have no fat finger errors, I'll call them, to make sure that we're not missing any data. I think recently there was that Amgen story that came out where a tab of missing data on Excel cost the company $12 billion. We want to avoid that as much as possible by bringing in all data automatically, and then ultimately deriving scientific insights. So being able to centralize data into one single platform where you can easily query data over a longer historical period of time or across multiple different types of workflows allows you to combine data sets that you normally were not able to even kind of compare in the past. But through centralizing it with Ganymede, you're able to come to faster decisions, more efficient decisions as well. So I think a huge thing that I've seen in my software career is like, does software actually change companies, right? I think we've seen a lot of fantastic softwares go out there that ultimately end up not being successful because adoption is a huge issue. I think if you can have all the best features in the world without having adoption, and essentially that means that it's worthless, right? So our paradigm at Ganymede is that we're not really trying to have people learn a new software. For a lot of scientists, this actually means that they will have to interact with software a lot less because a lot of these automated pipelines will be kicking in for them in the background. So things will automatically appear in informatics systems. It means that we can capture more complete data and more data, but actually spend less time in software itself. So really pairing data teams and IT teams' goals of having more robust data sets with scientists' goals of not having to spend as much time in Excel. And I'll pause here on the contact information and open it up for questions.
That's great, Gavin. Thanks. All right. Are there any questions for Gavin? I'd be curious to know if there are CMOs or other such groups in the room that see this as an intriguing tool. If there are some insights or some discussion points that are worth having, or maybe Gavin, if you'd like to share any kind of information about how Ganymede has interacted with CMOs, CDMOs as a way of advancing their workflows.
Yeah, certainly. So we've worked with CDMOs in the cell and gene therapy space a lot to really bring in all of their instruments again into that single data layer, and then be able to help them move towards GXP compliance as well. So a lot of the teams that we've worked with started in a non-GMP environment, but didn't have the data ready to go for full GMP compliance. So I think we are a huge first step towards getting there. And then also we offer validated solutions too. So we really help people bridge that gap from a non-validated into a validated environment. And having data be predictably parsed and analyzed really helps with regulatory submissions, with audits from the FDA, and being able to also then serve that data to clients automatically as well via a data pipeline is a huge unlock, right? And the turnaround time is instantaneous, down from either weeks, days, hours.
Mm-hmm, mm-hmm. Fantastic. Okay. Any questions in the chat? It appears as though we are good to go. Yeah, Gavin, I'll note that I am helping to lead a project with Schmidt Sciences Virtual Institute of Feedstocks for the Future, where we are in the process of gathering a wide variety of different kinds of data in the field as well as in the lab to evaluate the utility of agricultural residues as new kind of co-products or byproducts of the industry. And we will be definitely utilizing a variety of tools for managing a rapidly growing data lake of information so that we can perform both rational as well as non-intuitive types of model building and analysis. And so it's really great to see the kinds of things that are happening in the industry, such as at the companies that are here today, because it's a decided need that we ourselves are feeling in these large projects with lots of data over a long period of time. So this is great.
Yeah, I'm glad that resonates with you. And we did a initial pilot with part of LBL and JBEI to do a lot of this data analysis and integration for some of their chromatography machines. I can actually reshare my screen here and show you what that looks like. So that was something that they're working on as well. I can't share like too much about it, but what we can share is that we automated data capture coming from their HPLCs. We made that metadata easily accessible for them as well. So they were creating a lot of metadata and we're having trouble associating that with raw data. And the turnaround time for some of those data sets were quite large. But we were able, again, to bring down into mere minutes. And then we were able ultimately to track and associate relevant metadata across workflows and build really nice dashboards for them to be able to visualize all these different flows. So again, we were seeing that kind of human middleware as a starting point. Ultimately, though, we were bringing all of that into the cloud off of local PCs where it's prone to either going missing or just not being used as quickly as possible. Again, we were deploying an agent on that PC and connecting to an HPLC, bringing that into Ganymede and then structuring data automatically, being able to associate it with raw results, and then tabularizing and kind of democratizing that metadata for folks to be able to query.
And forgive me if I missed it, but Gavin, is there what kinds of QA/QC sorts of inferential conclusions is the software making, or is there more hard-coded kind of pass-fail gates that the user has to input?
Yeah, so a lot of those are hard-coded. We like to have the software be more, I would say, structured in that way. So again, all of that is user-defined code, so they're extremely customizable. We do have some kind of go-no-go decisions. We can basically split up different flows into, different data pipelines into multiple flows where there's a human in the loop. I think that's a lot of where that virtualization comes in as well, where we can then have a human in the loop do some manual QC, and then have automation take over for the rest.
Mm-hmm. Great. Yeah, I'll be curious to see with all of these types of software platforms, what the time savings and the opportunity cost kind of metrics look like as we begin to understand that more fully. It seems as though that 20% does seem like a really low kind of underestimate.
It is pretty conservative. We did a cost benefit analysis with one of our clients, Solugen, who works in the oil and gas industry, but does a lot of bioprocess. We've integrated several of their bioreactors, and we ended up saving them 3x their investment with us, over 2,500 hours. This is only really across like 5 different instruments. So even on a relatively small deployment, the time savings were tremendous. I think not only are the time savings great, but that also just frees up scientists to do more experimental design and kind of focus on other things. So not only is their hourly rate reflected, but they're actually outputting a lot more work. And then, of course, they were able to capture more time series data and more complex and complete data into their informatics system is Benchling. So talking to even just a couple of their teams and doing a handful of instruments, we were able to save them thousands of hours within months of deploying.
Mm-hmm. Right. Hours much better spent, like you say, doing things that that a human being can do and a robot cannot. So that's fantastic. Are there any other questions for Gavin and team while we have this time? Oh, Deepti.
So, Gavin, I'm curious to hear how much traction do you get internationally? Do you have international collaborators? Are there cultural differences in how we think about things in the US compared to the rest of the world? And how much of what we are thinking is actually, I don't want to say right or wrong, but are we in the right direction? Do you see what I mean?
I think partially I see what you mean. To be honest, we don't have that much international traction, but I think that's more of a facet of us not actively prospecting out into international markets. I would say the only one that we do is Canada, but I don't know if I would consider that like quantifiably different than the US as far as culture goes and geographical distribution. I think that we see, and we have like started to kind of break into EMEA a little bit. I think people are really looking towards kind of Lab 4.0 things and modernization of tech stacks where the cloud is being utilized for most steps in a process like data capture and analysis. I think even if you had talked about this 5 years ago, I think a lot of perception would be a lot different than it is now. I can kind of harp on my experience when I was at Benchling a few years ago, and I think it was a really tough time to be able to convince people that having a cloud-based ELN was okay. I think everyone would kind of chuckle at that now in 2024, almost 2025. So I think perception of products like this and the need for something like this is changing very, very quickly, and people are understanding that if you're making so much data, like you have to be able to utilize all of it because it's so valuable, and it really is the secret sauce for all these companies, right?
Yeah, that's good to hear because a while back I did iCore program, which is going around and asking people what they really need. And many operators push back on the automation of data collection because for them that was their creative part of the job to a certain extent, playing with Excel. That's how it was perceived. So it was unclear how we would be successful in getting the buy-in from operators who actually have to generate this data. So yeah, it's interesting that it's changing that quickly.
Yeah, and I think other comments on that. There's still, I think, some workflows where, again, people like to have a human in the loop, and I think that's going to be the case for a long time. I think even with the rise of AI, it's not going to fully replace decisions that are being made by humans anytime soon, right? So again, that's why we really lean on our virtualization product to be able to offer that to people to say, okay, let's automate all the obvious things that we can. Or even things that are less obvious, but if you really dig down, you can say, okay, we can actually automate this and have full confidence that we're doing the right thing, while still offering a couple of checkpoints or more complicated things where you need to have a bunch of different variables that change every single time that it doesn't really make sense to automate in a virtualized software in Ganymede, so that you're still creating contextual links to that data and being able to capture metadata where that's previously a black box in something like Excel.
Makes sense. Thank you.
Thank you.
My name is Jacob Pritt. I'm a data scientist with Mimetrics, and we are a company that does transcriptional data analysis. We have a multi-year record doing data analytics for ag, bio, and biotech companies. This includes research grants and collaborations with a variety of government organizations and universities. We have published research, and we have a toolkit of proprietary algorithms that combine mathematics, computation, and biology to perform time series transcriptional data analysis. So this plot shows an example of a plant, a single gene in a plant that's under infection by a fungus. And you can see just all the different gene dynamics that are going on with this gene at different points in the plant. And so doing a time series analysis allows us to really capture the trends and changes, much more than just taking one or two samples after some sort of change. So this allows us to improve performance, lets us help our clients improve performance and save time and money, both in strain development, process development, and in scale up. A typical project starts with experimental design, which is done in collaboration with the client. Data, metadata, and RNA-seq data is collected, and then we perform an analysis. As I mentioned, study design, we'll talk to a client, figure out what they are, what their goals are, what they're trying to learn, and establish experimental conditions and a sampling plan. Sample collection and processing can either be done by the client, or we have third-party partners who can do that. Then when we receive the data, we will clean the data, align and quantitate it, and then we'll do a data analysis. And this is done, again, it's an iterative process that's done in collaboration with the client. So one of the benefits of doing this RNA-seq analysis is that it captures data from the entire transcriptome. So it really allows us to do an exploratory analysis. So oftentimes our clients will have specific pathways or gene sets that they're interested in, and we'll go do the analysis, find out what we can learn. We'll do a preliminary report, and this will raise more questions. Oh, can we look into this pathway? How does this set of genes interact? And so with having that full transcriptome data, we can just keep exploring and answering questions. We typically do several different preliminary reports before we do our final report. And then afterwards, all the raw data, process data, all the plots and visualizations are all given to the client. So a few questions that we've helped people answer in the past, specifically from fermentations, are questions like, my metabolite production starts off really strong and then it tapers off towards the end of fermentation. Why is that happening? Or can I use a cheaper medium or cheaper experimental conditions? If I do that, how is that going to affect metabolite production? Another big question is, how do we balance cell proliferation and metabolite production to get optimal production? And later on in fermentation, how do we balance sort of stress response with metabolite production? And if these are genetically modified organisms, are the inserted or modified genes, are they working as expected? Are they constitutively expressed, or are they being turned on or off when they're supposed to? So RNA-seq really lets us move from looking at just phenotype to looking at the genotype and the gene expression that's underlying the changes that we can measure. So the top left here shows a time lapse of budding yeast cells. And then if we do RNA-seq, and look at the gene expression, we can see a beautiful cascade of gene expression like we see in the top right here. So in this plot, every vertical bar is a gene. Yellow bars mean that gene is being upregulated, and a blue bar means it's downregulated. You can see this wave of expression that goes through the genome over the course of a fermentation. And then once we have this gene expression data, our software can use it to predict the most probable gene regulations and generate a gene network, a most probable gene network like the one in the bottom right. Another example of this sort of gene cascade and the gene networks is can be seen in this visualization. This is from plants. You can see a few different waves of genes that are color coded. So we have this first wave of blue genes that are turned on early. These then activate the set of green genes. And one green gene at the top now then represses a lot of the blue early genes. And then several of those green genes in the bottom right then activate a late stage orange gene, which might be a target of interest. Another way to visualize the gene expression cascade is through heat maps like this one. So this is a heat map for the full yeast genome over the course of a fermentation, or actually this is just the first 8 hours of fermentation. So in these heat maps, each row is a gene and each column is a time point. Red bar means that gene is overexpressed at that time point, and a blue bar means that gene is underexpressed. The left heat map is just the raw unsorted gene list. And then when we sort the genes by their peak expression time, we can see a nice wave of expression starting in the top left and moving down to the bottom right. And almost all the genes are expressed at some point in the fermentation. As I mentioned, this covers the first 8 hours, but those first 4 columns are only 5 minutes apart. So you can see that there are a lot of genes that will spike for only a few minutes during fermentation. So this is another example where taking time series data can show you patterns or cell responses that you would miss if you were just taking 1 or 2 samples a few hours in. And once we have the overall gene heat maps, we like to filter down to different subsets of genes. Here are 2 examples of proliferative genes on the left and stress response genes on the right. And this can start to uncover some patterns. So in the growth genes on the left, you'll see that about half of them are turned on early on for an extended period, about the first hour or so. Whereas in the stress genes, in the plot on the right, you have a really sharp peak at 5 or 10 minutes that then drops off pretty quick. That's probably due to the shock of that fermentation. So if we're working with genes that have been modified and inserted genes, there are some different analyses we can do. We can look at sort of promoter specific expression. Another useful thing is to look at expression, average gene expression by location on the chromosome, to see if there are any effects from 4-dimensional chromosome structure or chromatin binding. So this plot shows the average expression of pericentric, subtelomeric genes and genes that are pretty centrally located on the chromosomes. So oftentimes we'll do experiments with multiple strains to compare. This was an experiment we did with 4 different strains, and an obvious question is, why are these strains producing more than these other strains? So these are 2 plots for 2 specific genes associated with enzyme production. On the left, you can see that for the green and blue strains, even at 40 plus hours, this gene is being still activated at a very high level, whereas the purple and red strains have dropped off their production of that gene, probably due to a stress response. The curve on the right is a little more complicated, not quite as simple, but in this example, you can see that the red strain is producing a lot of this enzyme sort of in the middle of fermentation, but it drops off towards the end, where in contrast, for example, the purple strain is much lower production in the middle, but it's still increasing towards the end of fermentation. We can also look at stress genes, like these are 2 stress genes shown here. You can see that in these 2 plots, the purple and the red strains are showing a higher activation of those stress genes at the very end of fermentation. So at this point, they are diverting resources to react to the stress. And as I mentioned before, this can be a sort of delicate balancing act where activating that stress response can help the cells survive longer, to produce more metabolite, but it can also divert resources from that metabolite production to react. And finally, I mentioned that we can generate these predicted gene networks. Typically, these will center around a few specific targets, and we'll look at the different transcription factors and see which are likely activators or repressors. In this case, we have this target, which is the blue curve in all three plots. And we found one potential activator and two potential repressors for that gene. So the top plot shows an activator, which is the red curve. And in the second half of this fermentation, when the blue, the target gene is activated, it's always preceded or right about the same time as a spike in that transcription factor. In contrast, we have two potential repressors on the bottom down there, which are elevated at a very high level early on in the fermentation, and the levels of the target don't come up until those repressors have a decreased level. So again, my name is Jacob Pritt. You have my email there, as well as our CEO, David Reed. Thank you for your time, and I'm happy to take any questions.
Okay, thank you, Jacob. Are there any quick questions in the couple of minutes that we have? Jacob, you referenced a couple of notable high-impact journals that you've published in with this work. I'm curious, in terms of impact, have you had an opportunity to kind of deploy these technologies in a way that you can discuss to give a sense of how groups have taken advantage of this rich data set?
I'm not sure sort of how specific I can get. We've done some work around sort of specific. We had a case where there was an inserted gene that was supposed to be constitutively expressed and was not, and we were able to establish the network, identify a repressor that was actually repressing that gene, and when that gene was knocked out, they then saw the expression that they wanted to see. So that's an example of one of our successes.
Mm-hmm. Right. Okay, great. All right, and so I think with that, please do take the opportunity to reach out to Jacob if desired. Jacob, feel free to add your contact info in the chat if you wish. And Teri, I think we can have you take it away for Polyplexus.
Got it. Thank you so much, James, for introducing me, and Deepti as well. Thanks for having us. I was going to just pull this back up. So I'm here representing Polyplexus. I have a few of my colleagues on the line as well. We appreciate you listening with us. We're developing what I would call the collaborative communication structure for scientific discovery. The Polyplexus solution began as a DARPA program. It increases the speed and the efficiency of first mile discovery by leveraging social connectedness architecture to surface and connect more ideas faster. And we're currently commercializing after being developed at DARPA originally. And the reason I think this is of interest for this group is so much of what you all know in your individual silos, if you were able to learn very quickly or connect very quickly with one another about these potential solutions, it would speed some breakthroughs in the field. Deepti and I have had the opportunity to work together a few times on some of these kinds of projects, and We've talked a lot about some of the knowledge that's out there that it just isn't accessible, so that people keep doing a lot of the same work over and over, as opposed to being able to share it and sort of move forward together. Of course, we're talking about the pre-competitive information. The competitive information stays competitive. But basically, we were built to accelerate science connections and knowledge exchange. And so what we do is we bundle a tested digital hub, and we can tell you some of the people that we've worked with and what we've done there, a built-in science and technology community, and then communication, facilitation, and data services, with the goal of delivering an efficient way to quickly assemble custom communities of interest across science and technology. So it's interdisciplinary, but they're usually, we create individual communities to investigate certain ideas. And so we see our partners as investors and innovators. We connect funding agencies, whether that's VC firms or government agencies, researchers, entrepreneurs, and ideas. We host targeted events, we host workshops, and we host idea and matching competitions, always with the goal of making it easier to horizon scan, to share knowledge, to find talent, and to source and create new ideas across geographies and disciplines. Our mission is to transform the collaboration of the first mile of innovation by connecting organizations efficiently and seamlessly. A lot of this connection architecture is missing in the sciences. It's pretty well developed in a lot of other areas. And the first mile of discovery tends to be a little more chaotic and a little less rigorous than the last mile of getting it to market. So the goal is to sort of get people in this place where they can talk and advance these ideas efficiently. We're a woman-owned small business with both a for-profit and a not-for-profit arm. And then we offer consultative approach. So we work with our partners to construct the programs that we deliver. And what I was going to say here is just our mission is more relevant than ever in this current climate. Ideas are coming fast and furious, breakthroughs are coming fast and furious, novel discovery is coming fast and furious. And yet the community is a little bit, it's unclear what's going to happen with some of the funding agencies. It's unclear where some of the areas where people have gotten together, what will happen with those. And our idea is that the time has come to create these more efficient ways of connecting. So the platform itself is highly customizable. I'll show you a few ways it was customized, but we can meet a variety of needs. We can attract and address related but diverse audiences simultaneously. We can quickly create focused topic exploration. We can enhance brand visibility within a highly qualified audience. We can establish, and I think here is where this is most important, a hub of knowledge exchange. And we can showcase funding opportunities for specific areas. So we did talk about how the fact that this was born in DARPA. DARPA had a lot of goals that they wanted to achieve with this. Most was making that first mile more efficient, but it was also in establishing and finding non-traditional performers or different performers and having better proposals. So over 30 different incubators, or they called them incubators, workshops. They doubled the number of submissions and they doubled their match rate by using this. And they ran these workshops in conjunction with regular BAAs. So they would have a BAA that went out and they would gather that, and then they would have this happening on Polyplexus at the same time. And one of the reasons that they could attract new proposers is there were no barriers to entry. People could come in. You didn't have to have a whole crew of people that knew the ins and outs of BAAs. We had global participation in every one of these incubators, but also because of the way it uses social architecture, program managers were able to guide the conversation in ways that allowed for a better match rate, and sometimes even just reformulated the question based on input. So because you can thumb up an idea, because you can thumb it down, because you can answer it in real time, this allowed them to greatly improve the match rate for the ideas they were looking at doing. So our pilot partners to date have included, besides DARPA, BARDA, the ARPA-E, ARPA-H, NGA, and foundations and private industry. And so as we structured this for commercialization, we've established a for-profit arm and a not-for-profit arm. And we also have the ability to create invite-only events so that they are not public, but privately invited, versus public workshops as well. So obviously DARPA, we wanted everything to be open, but we are able to create situations that are more private and more specific to a specific institution. And so the question is, how does it work? The first is it's the digital hub with participation architecture. Think of Reddit, think of Twitter or X now, but with an evidence-based component. So we have a feed where you can post DOIs and you can post pieces of evidence that just help people look and see, find different pieces of evidence. It's impossible to read everything that's being published. And so it's a bite-sized pieces of information with DOIs that have tagging abilities so that you can go ahead and look at those together. We then allow you to combine two pieces of evidence and create a conjecture. If this is true and this is true, this is potentially true. And that's really the feed portion of this. When we have an actual event, we have a Reddit type structure. We do support video as well, but have found that more people participate when it is in a Reddit-like thread, where people can go ahead and post additional pieces of evidence or additional questions within a discussion. So when we say a participation architecture, that's what we mean. We're trying to let people come at both the ideas and either the authors or the people who have posted those ideas in ways that facilitate evidence-based discussion. And then we activate a built-in community. So we currently have more than 7,000 community members on the platform. They have, you can see a way to create a profile. There's ways to follow them. There's ways to message them. There are ways to identify expertise. We also have a newsletter. We have multiple social channels. We have a podcast that comes and goes at different times. But this is really to support the community itself and say, hey, you can find someone that you need. You can find a coworker, and you'll see how that happens within an actual program. And the community attributes represent something like this. There's about 58% of our group are commercial businesses, entrepreneurs, research project managers, business dev, biz dev leads. About 28% hold positions in academia, and about 10% come from government and nonprofits, which includes 63 US government organizations and 196 global nonprofits. And so together, this is a very credentialed and interdisciplinary group that would be very difficult to replicate in other settings. So you would have to fly everybody in, you have to put them around the table. And so we can create these settings where you can get these people around the table to look at a specific issue or idea, or in your case, really look at other individuals who you want to be sharing information with that you may not know yet. And we can put you in the same room together to talk about the evidence and some of the things that people are finding. In terms of expertise, there are more than 1,500 areas of expertise represented right now. They include everything from public health to physics and neuroscience. And then in professional status, you're really looking at engineers, tech entrepreneurs, a lot of entrepreneurial people, research scientists, program directors. And I should point out that you are able to fill this information in on the platform, but you do not have to fill this. So this is a partial list of the people that are here that have volunteered some of this information. And I think the biggest thing here is really about engagement. So the idea here is not that this is a static repository and then you go in when you want to find a paper or you do. It's about engagement. It's about collaboration in the sciences. And so when we have workshops, we will have a huge percentage of the community working together and looking at these things together. We had a competition with ARPA-H around transforming health. And, at the time, 30% of our base was active on the platform at once, looking at these ideas and sometimes just looking at them, sometimes talking about them. The community has uploaded more than almost 11,000 individual pieces of evidence that they've put onto the platform. We know from a lot of discussion that the networking is extremely valuable, and people have either made hires based on who they've met on this because there's an active demonstration of how someone thinks, or they've collaborated on a project together. So at the end of the ARPA-H presentation, we did hear back from some people at one of the national labs saying, hey, we reached out to the winners because we think we can work with them to create a different formulation of this. And then connection and reach. So when we're not on the platform and we're sending out newsletters and we're producing other pieces of content, we have a huge engagement reach. And our community really spans 6 continents and nearly all 50 states. I'm sorry. So we really have a wide reach. And when we set up an event that's in a specialty, we then engage that many more people who are interested in that specialty. And then finally, we have communication, facilitation, and data services, and that's what our teams provide. So this is not the kind of platform where you come in and it's going to just be automated like Asana or something like that and try to guess what you want to do. We're actually tailoring events and we're facilitating the communication of these events for people, and then we're providing the data services on the back end. So this event was called Evidence Madness. It was a competition to identify what was considered seminal scientific evidence published within the last 5 years that participants thought would lead to new transformative industries by 2040. It was set up, this was actually an idea by a sponsor to set it up almost like a March Madness bracket. So you would have 4 different divisions: life sciences, computer sciences, physics, and I forget what the other one was, chemistry. And then people submitted their ideas as to what was the seminal evidence. Some subject matter experts went and vetted those and filled out the brackets, and then it was open to voting and commenting. So you can see we had 63 organizations represented, 44 universities that ranged from MIT to the University of Ljubljana, and 6,500 votes to create a winner, to say, hey, this is the idea that we think is the most likely to lead to transformative industry by 2040. And so, you know, when people put in evidence from all over Nature, IEEE, MIT Press, and more than 40 countries participated in this. So this is what we're talking about when we say we design a program, we design an effort, whether that effort is an ongoing effort that goes for a long time or a short effort that goes for 3 weeks. We design that effort, we communicate that effort, we solicit experts to join us in the discussion, and then we provide data services on the back end. So what we're really talking about here is a platform for fast, efficient connection in the sciences. We want to allow people to move faster with focused idea and talent searches. We want to gather custom communities of interdisciplinary researchers around areas of interest. We want to create meaningful interactions, so not just doom scrolling, content and community in highly specialized fields. And we want to accelerate recruiting, outside expert engagement, and S&T outreach with efficient real-time events. So the question is, what does this look like? And I'm going to take you through a case study, but I think I have to stop sharing and come back and reshare the other tab. And see if I can do this. And can you all see that? James?
Yes.
Okay, great, just making sure. So this is just a design, so you can just quickly see a quick way that ARPA-H used the platform for their goals. So they had this idea that they wanted to increase brand awareness for the agency on their first anniversary, but they also wanted to survey the field and understand what a broad group of health constituencies across the United States were caring about. So they wanted to build the base of a custom online community and identify potential program managers and programs through online interactions. And it began with a simple question: do you have an idea that could transform healthcare? We then had a branded login process so that when you went into Polyplexus, you knew you were in the ARPA-H event. When you sign into Polyplexus, it's very simple. You just create an account, which means you add your email address, or you can sign in with your LinkedIn profile. And then once you were there, there was a portal and there were a series of tabs. And the first tab was really the invitation to submit. And what they designed was a 6-question submission form that really mirrored the Heilmeyer effort, and invited you to say, hey, if you have a lot of competitive information and you want to go through the BAA door, go through the BAA door. But if you just have a really good idea and you think this is possible to do, we want to know how it's being done today, what you think is possible, and what the evidence suggests is out there that suggests that this is in fact possible. So it was just 6 questions to be considered for the program. There was a 10-day submission period. During that period, we had 35,000 views, visitors from all 50 states, and more than 500 submissions across the ARPA-H focus areas. And these submissions came from the broad group of people: universities, major health organizations like MGH and Mayo, privately held startups, public companies, and foundations. The partial list had some of the usual suspects, so you would see a Duke or a Harvard, but it also had people like the University of Toledo and the Illinois Institute of Technology and the American Vein and Lymphatic Society, as well as a bunch of startups like Seleno and BioTrillion. Leapfrog was there, IBM was there. So we had a very broad group of people who submitted and participated. Once we got those 500 ideas, the subject matter experts in health evaluated the ideas, and it was really sort of an abbreviated way to do some peer review, and said, hey, these all are valid and worthy of consideration. We filled out a bracket of 64 different ideas, and we published those brackets and left them open for discussion and voting. And when I talk to you about the way it works like Reddit, under every one of these things, and you can find this on the site now, but under every one of these ideas, so the little green boxes, the little pink boxes, etc., that's a submission. And under that submission is a conversation about that submission. So people are saying, hey, I think that is possible. I've seen that done. Here's another study that replicates this, or here's another idea that's related to this. And every few days, we would have the voting, and based on the voting, one of the ideas would advance. ARPA-H reserved the right to say that if they needed to reverse the voting and they thought the public was wrong, they could, but they did not exercise that during the whole program. And we eventually got to a finals that had two amazing ideas on it. And one was one drug for all cancers, and the other one was your cells, your cure, with personalized stem cell technologies. There were 6 rounds of voting and discussion, hundreds of comments on the entries. 8 semifinalists came from various portions of the country. This particular event was limited to the United States. End to end, it was 7 weeks, and provided them with clear differentiation, but also created a community with direct access and dialogue across these ideas, and a lot of talent and idea identification. So in the short term, the champion idea was one drug for all cancers. There were, I think we talked about it. Sorry? We talked about
Okay, thanks.
Hi, David. We talked about intangible benefits and tangible benefits, but we were really much more interested in the long-term results. And one of the long-term results was Seleno, which was the runner-up in the competition, as a direct result of its participation in the vetting of its idea, the surfacing of its idea in this competition. They were awarded $25 million in research funding from ARPA-H for this new project. We talked about the community. This is a really big deal. Here's what Selena said: been very motivated by seeing how many people have been interested in submitting ideas and commenting, engaging in debate and discussion, not just the scientific community in this case, but all across the country, engaging and dreaming big about how we tackle health challenges. And just the connection, inspiring to see all the efforts that have been going on. Really pleased by the conversations we've been able to have with the number of people that have engaged with us. I'm hoping in the future these people can become collaborators. And the third one this content was fantastic fun, and my company's in negotiations with the winners to help them formulate a different drug candidate for inhalational use. It's a great opportunity for both of us and for the ultimate recipients of the drug. And it would never have occurred if it weren't for this particular contest or for Polyplexus in general. This is what we're going for. This idea that we can together this information by putting it adjacent to, putting people adjacent to one another and putting the information and the evidence out there, either publicly or privately to a specific group, that we can advance that first mile more quickly than it's currently being advanced. So let me see if I can get back out here and then just give you my contact information. So if I can do this. So what we really are trying to do is promote interdisciplinary evidence-based conversation and provide easy access to ideas, researchers, research funders, and talent. Deepti and I have had several discussions about, in your field, easy access to failures also, some of the things that didn't work so that you don't have to replicate these studies again and again. Efficient and accelerated workshopping and discovery, and configurable, sustainable, expanding ecosystem that's focused on information sharing. Again, in the pre-competitive phase, with publications that are out there. Our core services are workshops and events, breakout sessions and idea challenges, knowledge repositories, data, and communication strategies. And our plea would just be, let's work together in an evidence-based environment, because the world needs these better ideas faster. And here is our contact information.
Fantastic. Excuse me, Teri, thank you so much. This is great. Okay, are there any questions for Teri? We have a good 6 or 7 minutes to play with. Yes, Deepti.
Teri, thank you very much for giving this talk. I wanted to hone in on a couple of things. So one of the things that we see happen in industrial biotech, and maybe that's slowed down a little bit for the past year or two as we are going through a correction, but we had a lot of turnover at many of the companies at the ABPDU and many organizations. Even small companies, after 2-3 years, they take a lot of decisions, and 3 years later, they don't know why they're doing certain workflows a certain way, and they're reinventing the process. Is there anything Polyplexus can do for people within their small group of community or within their company, small company, over a period of time? I totally see the value proposition in communicating across companies and with funding agencies, but what can we do for small companies? That's the question here.
Sure. So creating a knowledge repository for an actual individual entity, we definitely can do that as well. I think the power of a platform is always when you take those individual things and connect them. But you definitely could create a group that's just for a company within this platform as well. So we really work on the configurability of this so that it makes sense. There was also some other things came up on the smaller company level. So one group was saying, hey, we regularly publish what we think is going to happen in the future of their area of inquiry, and would like to go back to our reports and ask the field, how close has this happened? And where are we succeeding and where we thought we were going to succeed, and where the technology is really pushing forward and where aren't they? And so there's a lot of different ways to use this when you think about it as groups of people and evolving knowledge. It's sort of how we're going to connect those dots on the platform. But all the basic functionalities are there to do that.
So that's a very good thought, because one of the things that we are seeing now is an emergence of self-driving labs, right? So AI will design the experiments, and then AI will execute the experiments, and with automation, we'll execute the experiments. One of the issues with that is AI doesn't necessarily know the difference between a good experiment and a bad one. And this came up at a National Academies workshop where someone challenged that if you asked AI, it would tell you that 96% of people who died in an air crash drank milk in the morning. So one of the experiments to do would be to, feed milk and see if they die in an air crash, right? So maybe 50% of the experiments that are being conducted through self-driving labs are not necessarily useful experiments, but automation can help us, right, fast. The problem there is, the question here is, how do we compare? Right now, we do experiments, we do analysis using AI or general, simple statistics. But then we take some decisions and then we move forward and design more experiments. We are not keeping track of that decision-making process very well. So there is no way to compare if AI is actually deciding better than humans. So I don't know if you've all thought about that, if there's
We
any.
have talked about this, and so we haven't talked about self-driving labs per se, but we have talked about creating some AI agents as profiles. And so letting the community, so really doing that human in the loop on the front end of the process. So let them propose evidence, let them propose conjectures, and let the community say, off the mark, or no, actually, this is really helpful. And using it in that way to look at the contributions, right? More upfront, not in the back end, not in the middle, which is usually not transparent to the community. So really because it's an expert community, we can do the human in loop on the front end. So we have talked a little bit about that. I was at a couple of seminars talking about how we would do that. I mean, there's obviously ways to integrate AI into this. We're making some suggestions and creating stuff, but I think it would be much more interesting to actually put it front and center and say, here's the result. And it would be really interesting also to think about some kind of a workshop that looked at self-driving lab results and created a larger community to then evaluate those results, either for further learning or just to validate or invalidate the findings.
Yes, that makes sense. Thank you.
So Teri, while we've been sitting here, I've been exploring the platform, and you'll have to forgive me if I missed any kind of tips and suggestions, but are there things that you would recommend right off the bat for somebody who has just jumped on the platform and wants to kind of understand it a little more?
Yeah, I think create a little bit of evidence, look around, see if you might pull up a creative conjecture that goes together. But I think what's most interesting, and where people would tell me that they lose themselves all the time, is we archive all the public events. So if you go into the incubators or workshops tab, and you can go see all of them, even if they're closed and they've ended, but you can go in, so you can find that ARPA-H event, and you can just go and do it. So we've been told, is a lot of analysts go in afterwards and they look at it to try to figure out what were people proposing and how were they thinking about it and how were they connecting the ideas. So that's how I would say browse that. It's intellectually so interesting. And you can go into some deep dives and find some really interesting things. That's where I would start. And then I would go to the feed and add some evidence.
Great. All right. Will do.
Well, thank you very much. We appreciate the opportunity to be here and certainly respect all of your work.
Thank you, Teri. All right. So with that, I think we can move to David Hong of Synonym.
Thanks, James. Can everyone hear me?
Yes.
Great. I'm just going to start sharing my screen. And then also thank you, Deepti and Leah, for organizing this event. Really appreciate the opportunity to speak. Okay. So hi, everyone. My name is David Hong. I'm the Senior Director of Business Development over at Synonym. I spent most of my career at DuPont, where over the 12 years there, it was GenCore, DuPont, Dow, now IFF. I spent most of my time where my business unit was selling enzymes to the cleaning industry, so proteases, amylases, mayonnases, you name it, to companies like P&G, Unilever, Reckitt Benckiser. From there, I went to Culture Biosciences, where it was a startup many of you are probably familiar with. They helped outsource bench scale bioprocess experiments. And then from there, I went to Perfect Day. Perfect Day, as many of you know, commercializes and sells beta-lactoglobulin or a whey protein via precision fermentation. I didn't work on the whey protein aspect itself. I actually helped them build and launch Nth.bio, which was a biology as a service platform. So any bench scale bioprocess experiments, piloting and manufacturing, all the way up to 660 kL tanks, was under my watch. And it was a great time over there, working with companies, seeing if they can get to their commercial scale capacities in my tanks. But I was limited to just those assets that were, under my umbrella. And now over at Synonym, I'm able to branch out and unlock this whole network across the globe to see if anyone can utilize any of those assets and facilitate these connections. And that's what I'll be talking about today under the platform solutions. Next. So getting a bioproduct from lab to shelf is a challenge, and then Synonym can be your partner and make your life easier throughout this entire journey. Our platform solutions are software-enabled products that facilitate your journey from R&D all the way to commercialization. And the products that we're offering, that I'll talk about today, are Scalar as well as Match, powered by Capacitor. I won't spend too much time today on Scalar because my colleague Quinton, who's actually going right after me, is going to cover that right after this presentation. Just a little tidbit is that Scalar is a dynamic and robust TA, and you can also think of it as a decision engine or a decision platform. Techno-economic analyses or TAs provide a systematic as well as rigorous evaluation of the economic viability of a technology. And TAs are a key resources that help businesses inform decision-making to maximize your upside, minimize your potential costs and your risks. And you can't or shouldn't be making informed data-backed business decisions without something like this. Here's a peek at the deliverables. And I'm not going to spend any more time on Scalar because, like I mentioned, Quinton, right after me, will go in depth on this topic. And I'll also unveil an exciting update on how we deliver TAs at Synonym. Moving on to CMO Match, powered by Capacitor. Synonym Match helps companies find and enable fermentation production capacity to support any and all of your scale-up needs. Even if you know where to go and which CMOs you want to talk to or which ones you're interested in, going from cold outreach all the way to establishing an NDA can still take quite some time, depending on the amount of time it takes, to get through the legal revisions. And this is true for every single CMO conversation you want to have. From there, you'll have to still have after an M&A is executed, you'll still have to have your initial tech transfer, have a series of calls to help the CMO better understand your process, all the requirements, and then from there, they'll build a quotation from you. And even if you've made it this far, there's still a lot of problems that you can run into along this journey to commercialization. And many CMOs conversations can end up being dead ends. Explaining your process during the tech transfer can be filled with tons of miscommunications and barriers due to the geography as well as the time differences from you, as well as just tech transfer is just very hard to do across multiple sites, multiple teams, and multiple facilities. Once you get those quotations, they can also be out of budget. There might also be CapEx requirements and surprises due to the mismatches from when you started this conversation and process. And CMO contracts are also come in many different forms and flavors. They draft their agreements in many different ways, and converting all of those quotations received into an apples to apples comparison can be difficult when you're receiving quotes that are by batch or campaign, by time and facility, cost plus, dollars per kg product, et cetera. And you can really dedicate a whole FTE, for a year or more, just going through this process over and over again for each CMO. So Synonym aims to make this whole process easier, and we want to conduct this request for proposal or quotation for you. So we want to cut out multiple steps, but also streamline and scale the search with parallel conversations on your behalf. We'll have an MNDA in place with your company, as well as a lot of the CMOs and production capacity providers that are on our network. After intaking your process, we'll query our capacitor directory and filter all the facilities by tank size, certifications, organisms, region, DSP unit ops, maybe even budget if that's available, and any other requirements that you might have. From there, after querying that database and directory, we'll have a short list of potential partners to produce your bio products. And then we won't even stop there. We'll create a tech transfer document and then request those quotations from all the CMOs and facilities that we've identified that are potential partners, that are CMOs, but also facilities that are not traditional CMOs. You know, we can even open up other conversations about co-locations, brownfields or greenfields, you know, whatever might be best strategically for your project or your company. From there, we'll deliver a report of facility profiles, quotes, and even facilitate introductions. This service should expedite the whole process. And, you know, it's not meant to be a cheat code, but it's meant to fast forward you. And then you'll still have the need for deeper technical conversations and site visits before you do everything, but you'll get to those steps quicker and you'll waste a lot less time. Our screening and qualification sets up any meetings that you have with these CMOs and sets you up with a better chance for success, and which hopefully in the end is a production partner that you trust and you'll be happy with. By leveraging our capacitor directories database, we can provide an initial assessment of production partners for you with just a few pieces of information. And, you know, this initial screening just starts with your desired capacity, a manufacturing grade, region, product type, organism, just DSP unit ops, and then we'll be able to return with a quick list, as I mentioned. And all of this is made possible by Capacitor, which we believe is the most comprehensive directory in the entire world. And our service will allow you to focus on other mission critical priorities while we take this on for you. Anyone that has taken on this search knows how challenging, time consuming, and you have even heard disastrous as a descriptor for this whole process. And I'm sure many of you have visited the Capacitor.bio website, but if you haven't, our directory spans close to 300 facilities in over 40 countries with over 50 million liters of fermentation capacity at your disposal. And this is just what we publicize. In our network, we actually have an order of magnitude larger than this. If you do want to get started, onboarding is quick. And once a project is kicked off, we expect quotes to start flying in within just a few weeks, maybe a month, depending on how responsive or how difficult your search is, you know, depending on if there's any special requirements that you're looking for that make this task not as straightforward. So if you're interested, please send us an email at platform@Synonym.bio to learn more. And I think we have still 5 minutes, so let me know if there's any questions.
All right, David, thank you so much. One quick question. You quickly alluded to an order of magnitude greater capacity relative to the facilities in your database versus what you screened. Curious, what is the difference there?
Yeah. So a lot of the facilities that are listed on Capacitor right now are traditional CMOs. You know, they're tollers, they just want to be a producer. There's a lot of other companies or facilities or companies, that see us as a industry leader, a thought leader in this space. And they've actually come to us and they've said, hey, I have this asset, this facility, this site that I only utilize for half the year. You know, the other half of the year, I have open capacity. Do you know anyone that, you know, could use this or is a good match for this process? These are the organisms, you know, they're really coming to us and they're asking us to help them find a partner or someone to fill up that space that strategically makes sense with their location or what they're looking for. And so, like I said, not all those facilities want to be traditional tollers for most of the time, so they don't want to publicize their assets and their information, but we do have connections there. And that's kind of like our secret sauce or magic behind.
So it sounds like 90% of the capacity that you reference actually is in that form versus the toller.
I can't speak to the actual numbers, but I think that's at least double or triple the amount that's publicized on our website.
I see. Oh, we have a question from Chandrith Thakur. Thanks, David. How does scalar capacitor platforms work with startups or academic researchers who might have limited resources or infrastructure?
Yeah, so that's the great thing about this, because even for startups, sorry about that. You have to start somewhere, right? Whether you're going from bench scale, or starting to do those first pilot and demo batches, maybe not demo, the pilots. This service spans all the way from bench scale, Ambers, even biolector service providers, all the way up to production scale commercial manufacturing. So we can help any step of the way. I'd say most of the more established companies, that have been in the space producing at the pilot and demo facilities, they know where to turn to, who to go to, or have connections to these traditional CMOs. But a lot of the other newer companies don't have these connections. They don't know where to turn to. And like I said, that whole process is very opaque. You're seeing hundreds of thousands of dollars, maybe millions of dollars difference in quotations, and you're not quite sure why or how to even start or how to discern any of this. And so we can really help facilitate that and guide you, hold your hand throughout this whole process at any stage or scale. Obviously the smaller ones are going to be lower cost, and that's what you need, and that's what we want to do, is just help rise the tide and help everyone get to commercialization faster and in the best way possible.
Okay, great. All right, we have a question from Anna, who is asking, do you also look at CMOs that could support chemical reactions, such as for making derivatives?
Yeah, so I would say that the majority of the facilities here are going to be for precision fermentation, but we do have a lot of queries for biomass. We have a lot of different flavors of CMOs. I say a lot of companies are very buttoned up with their upstream. They have question marks when it comes to downstream, or maybe they have a DSP that is not scalable, but they just need to get their product first, and they need a partner to optimize their DSP or to figure out what it is to get to the next steps. And so we have a lot of facilities that specialize as DSP shops or that can help you get there. There's going to be challenges with some companies that need methanol or other solvents and any types of special requirements, as I mentioned before. And really each process is going to be unique, each search is going to be unique, and we're able to at least listen to you and intake your process and let you know if it's something we can take on. And I would say that with a certain degree of comfort and success, we would be able to at least start that search for you.
Mm-hmm. Okay, great. Thank you, David. We also have a question from Alexandra, who is wondering, how frequently is the database updated?
This is updated in real time. So a lot of this is self-reported from the CMOs themselves to make sure that they're up to date. So I don't think especially for the smaller ones, that there's so many facilities all over the world. For the major players, we have regular contact with. Like I said, we have the MNDs in place. We have open lines of communication. We're funneling projects to them all the time. And so we have an updated list of their DSP unit ops and their tank sizes and any updates and CapEx projects that they're taking on. For the smaller ones, it's really up to those other companies to self-report. But we might in the future run through and see to make sure everything's up to date to better.
Right. Because I could see information becoming pretty stale pretty quickly and people forgetting to check it. And
I would say that there's a couple other, similar services that I've seen that might be the case, but I would say that we have a pretty active community here. People want to make sure that their assets are up to date, and so I'm confident that, you get a good representation, at least at a starting point.
excellent. Okay, so with that, I think we can turn it over to your colleague, David. Quinton,
All right.
also Synonym.
Thank you.
Thank you.
Thank you for that.
All right, well, I'll just get right into it. I appreciate you guys having me on and all of the work that's required to put this together. I'm Quinton Cohn. I'm on the Synonym platform team. My background is in engineering, and more recently, I've worked at companies in assessing biomanufacturing solutions for industrial biotech technologies, particularly focused on the economic feasibility for the technologies and the chances to get them to scale. And so Synonym as a whole is a partner for biomanufacturing scale-up services, but Synonym platform focuses more in on data-enabled and software-enabled services that we offer to our clients to help them reach their commercial scale in the most efficient manner. The two products under Synonym platform consist of a techno-economic analysis tool called Scalar and CMO Match, which David just went over. On this presentation, I'll be focused more on the techno-economic analysis tool. So what is a TEA, or a techno-economic analysis? TEA for short. TEAs can provide insight into what your production cost can be at any scale in your process. TEAs can also incorporate large data sets and variables, which can help you manage KPIs and resources more efficiently at any scale, as you go from maybe a literature study to a lab scale to pilot and hopefully eventually commercialization. They can also be used to attract investors, or from the investor's perspective, deem technologies feasibility as they're contributing capital into these technologies. Some of the ways that Synonym's TEAs work differently: for starters, it is a cloud-based software tool. So it's dissimilar to traditional Excel modeling that requires large amounts of process engineering labor. It's growth-oriented, it's customizable, which means at any point in your process, you can come into your Synonym Scalar TEA and update it, and your outputs will automatically update. We offer the ability to adjust and change at any given time over 70 different parameters and inputs. And it can also provide dashboards for resource allocation or presenting to boards or investors or whatever you might need to be doing with it. Some of the key deliverables, which I'll get into the details in later slides, are listed right here. All of our TEAs will come with mass energy balance equations, tornado diagrams, which can be used for sensitivity analyses, production cost estimates, obviously, and then breakdowns of your cost of goods sold, as well as your capital expenditure. Our TEAs are very customizable and malleable to your needs. And typically when we onboard a new customer, we give them essentially a menu to choose from. You can select as many different features as you need, but these features are listed briefly here. I'll get into it later, but some of the things that might be of note are your CMO cost comparison. That would be what your cost is to contract manufacture your process rather than building a greenfield site of your own. We do offer carbon intensity reduction analyses, which would be your CO2 equivalent per kilogram of product, and also some tools around how to reduce cost as you scale up. We also offer ways to analyze and compare different decisions that you might have as you scale up, such as location. Do I want to manufacture in the US or India or Europe, whatever it might be, and then feedstock. So as you're looking at different feedstocks to produce the lowest possible production cost for your given process, you're allowed to compare all of these different features. Downstream processing is similar, as well as fermentation type and product grade. And now I'll get into the methodology and how we actually produce the robustness of these TEAs. So we always start by calculating our capital costs. This would be done by providing a block flow diagram. If you don't have a block flow diagram, it's okay. We can build these TEAs with something as small as a literature study and a list of downstream processing unit operations all the way up through commercial scale and FEL 1, 2, or 3. Within each of these blocks will give us the information to understand what pieces of equipment we need in each one of these areas in the process, and also the mass energy balance that's contributing to those pieces of equipment. So all the energy and the conversion metrics associated with each block and each piece of equipment, we calculate that, we tie it all together, and ultimately we come up with a sized equipment list. So your sized equipment list will be every single piece of equipment for the given size needed to reach your target output in a year. as well as the price for each piece of those equipment. And when all that is summed up, you will have your price equipment list. Underneath these areas and these pieces of equipment will obviously be real estate. And so we also calculate the area underneath the plant, what that will cost you. Different building areas have different costs associated with them, and we make sure to adjust for that accordingly. Once you have your priced equipment list and your land cost, we throw in some more cost in there, such as your construction directs and indirects. These are basically costs that you can't avoid. They will be necessary in building a greenfield plant. And we roll this all up into a CapEx breakdown, which you will have access to. You can see how each variable is contributing to your total installed cost. And that's essentially your CapEx. Along with the block flow diagram and the mass energy balance, we get into our operating costs, which is the next piece of the puzzle in deciding if something is economically feasible or viable. The operating expense is done by figuring out what your raw material cost is, that's your feedstock, plus your media, any chemicals you might need or consumables, and then your utilities, your water, your electricity, natural gas, etc. Those would be your variable COGS. And then once you add in your labor, you would get your total cost of goods sold. I won't get into this in detail, but we do have very robust ways of calculating electrical usage. This can be good for estimating carbon intensity, but also cost, as well as your wastewater. All the water that's used and disposed of in the plant will be calculated and handed to you in your model. With the labor model assumptions, the way we think about this is depending on the plant size and the operations used in your plant, we'll give you a labor model, and each one of these blocks in the labor model will have costs associated with them. This can be different for different geographies as well as different scales and different processes. And this will also flex as you adjust your model. Another thing that we always include is your financing costs. We understand that capital is not free. And so depending on the capital necessary to build your plant, we use a debt equity model with targeted levered IRR and cost of capital to understand how much financing cost will be and what it will be as a percentage of your total cost. Once we have that, we will give you and we calculate your breakdown. This will be similar to your CapEx breakdown. It'll give you every single variable itemized and what contribution it's providing to your total production cost. As you can see, capital service is sometimes overlooked in a lot of models, and it can be quite high in some cases, as the capital needed is oftentimes in the hundreds of millions of dollars. With every single variable, we calculate uncertainty and sensitivity. This would be done by providing a baseline where we start, and then we have upside and downside scenarios. You will be able to, in your model, you can adjust the entire model to an upside scenario or a downside scenario, and see kind of in what range you're looking at reasonably being able to fall within if you do scale up to commercial scale. And then breaking it down even further, you do have the ability to adjust the whole model, but we also offer tornado diagrams. This would give you an idea of how each individual variable will flex your production costs as you go from the upside cases to the downside cases. This can be very insightful for R&D teams to look at and how they expend their resources, because you understand what you need to optimize for what's going to move the needle the quickest and ultimately drive the most value. Similarly, we offer waterfall charts. This is great for road mapping. If you can imagine at the lab scale, you might be calculating your cost, and it could be much higher than what you're promising to investors or what you would need to sell at in order to succeed for the given target molecule. this chart will give you an idea of where you are today and what would be needed in each one of these categories to reach your desired production cost. And the last thing I'll hit on before questions would be calculating your emissions. The way we do that is leveraging the mass energy balance again, and then we apply emissions factors, which will give you your estimated carbon emissions. It'll be your CO2 equivalent per kilogram of product. And with that, I'll open it up to questions. If you are interested in learning more or diving deeper into this, please email me at Quinton@Synonym.bio or Synonym.bio to get started. And then I guess I will, I'll just show you a brief look at what the software will actually look at. This is a demo environment, and we can go into it further if you schedule a call or if you're interested in learning more, but I'll open it up for questions now.
Okay, Quinton, thank you so much. Any immediate questions? What are your sources for carbon emissions factor? Comes from Antoine.
Yeah, we use the EPA GREET model to estimate the emissions. These are basically factors that are applied to every single item in the process, but we are also flexible with this. Like if you have done an LCA and you have metrics that you would like us to use, we can use those as well.
Okay. Let's see here. Human Parhitskar has a couple of questions. Does the platform support budget allocation for human resources and the expertise needed to scale up? Is one question.
I would assume, yeah, I would assume you're talking about the labor model. We do actually, we do have different factors in the roadmap. We apply different factors at various points in your production scale up journey. So it won't be calculated in a super robust manner, but we put in factors to best estimate the inefficiencies you will experience as you scale up. That would be a good example of one. Another good example will be that you would probably be only at 20% capacity in your first couple of quarters, and then you might experience some failed batches. So we do our best to calculate to the most accurate manner we can any of the inefficiencies that come along with scaling up your process.
Mm-hmm. Okay, great. And a second question from Human is, would it be possible to get feedback on lab scale equipment design, experimental design? Yeah, it's obviously so much of this starts out with very small scale equipment.
Yeah, and that's a great point. And we do offer that, especially in the comparison tools. If you're looking at several different processes at a lab scale, as long as you've run a fermentation run, we can compare those processes to each other and kind of look at what a hypothetical commercial scale would look like. But the TEA does enlighten feedback on lab scale experiment design and can give you a good direction to go as your next move.
Okay. All right, and Chandresh has a second question, and that is, how does scalar TEA approach differ from sort of the traditional tools? You can think of SuperPro or Aspen.
Yeah, I guess every tool has their pros and cons, and some of these tools are kind of broad market tools for multiple industries. Scalar was particularly designed for the biomanufacturing industry. And so every single feature set in there and all of our pricing lists that are updated every month are specifically suited for this industry, as well as just the feel and the user experience. I think if you get into the demo, you could probably see the differences a lot easier. But it basically gives you your roadmap, your bio-MRL journey of what all these different stages will cost and where to point you when allocating resources that a lot of these softwares will not provide.
Got it. Okay, very helpful. Okay, so with that, I think we can conclude session 1.
Welcome back, everyone. This is our second session for today, and second and last for today, but second out of the 5 this year's expo. So for this session, Eric Sundstrom will be our moderator. Eric, please introduce yourself and get going. Yeah, all yours.
Welcome back, everyone. I'm Eric Sundstrom. I'm a staff scientist at ABPDU, co-lead the fermentation team. And just a reminder, please feel free to put your questions in the chat as we go through our talks. Our first talk today will be, in this session, Apostolos Zournas from Lawrence Berkeley Lab to discuss media optimization for fermentation. So, Apostolos, you can take it away.
So today I will be talking to you about media optimization through machine learning. We have talked about in the previous session about different ways where we can store data and how to sort of automate that and reduce a lot of the work required for that. And this is the part where we use the data for process optimization. So as a disclaimer, there is a preprint on this work and we are under review right now. And why do we want to do media optimization? Usually what has been done, at least for the past few decades, is you have one parameter that you want to optimize. In this case, it could be C/N ratio. You select your nitrogen source. Then you go run a few experiments in particular concentrations, and then you explore a very small amount of the phase space. Whereas when you have machine learning, you can test a lot of things, especially coupled with automation. You can test a lot of things in parallel within the TBTL cycle paradigm, and you could explore the phase space a lot more efficiently and get results that you wouldn't necessarily expect. So this is a case study on maximizing flaveolin through media optimization. And the reason why we chose flaveolin is because it uses only malonyl-CoA as a precursor, which is a precursor for many other interesting things. It can be measured optically by measuring absorbance 340, and is only one step away from the central carbon metabolism, which is generally quite robust. So this would be a good case study. Changing the metabolism of that part through media would be very interesting so that we can have drastic changes. And we use the automated recommendation tool, or ART, where this is a machine learning model that will generate recommendations. And the way that it does this is it takes in some data, where in this case, the input data was the concentrations of different media compositions. In this case, we had 12 or 13 media components. We have the response, which in our case is absorbance 340 as a proxy for flaveolin. And we generate a predictive model, and then we query this model for the next TBTL cycle to either give us the maximum predicted titer or the maximum uncertainty, since the model can also give us that. And we either explore through maximizing uncertainty so that we inform the model as best as we can, or we try to get the maximum titer. And in this iterative manner, we can see here in every cycle, we get new recommendations. So initially, we had 2 TBTL cycles that we selected data through classic design of experiment techniques, namely light and hypercube sampling. We then fed that data into ART, and we got some recommendations. And then by fitting all of this data into ART, we got new recommendations and so on. In this case, what happened is that our best performer, the highest flaveolin titer, the media that gave the highest flaveolin titer, was at the 3rd TBTL cycle, or the first learning cycle that we did. And we did this 3 times. We started from MOPS minimal media all 3 times, and we performed 2 campaigns maximizing titer and 1 maximizing yield. And for this, we got significant improvements where we, and we managed to increase titer by 60 to 70%, and we managed to increase yield by 350%. One very interesting thing that happened is that for all three campaigns, the algorithm converged in the same region, which can be seen through the PCA over here. And you can see that as we go more to lighter colors, we go to higher DBDL, to further DBDL cycles. And as we go to more red colors over here, we go to higher titers or higher performance. So this region was where all three campaigns converged. And also this is where most of our high-performing media designs are. Something that was very interesting in what we saw is that sodium chloride at very high concentrations very much enhanced flaviolin production. So when we had when we added concentrations as high as 480 millimolar, which for Pseudomonas putida, which was the organism that we were using, is quite high. We saw that this is where we got the maximum flaviolin titers. Sorry, this was for the titer maximization. And then when we did yield maximization, we saw that, again, sodium chloride was a very important factor. But of course, because we're divided by glucose concentration, in this case, glucose was the most important factor. These are the data from our from campaign 3, where we were maximizing yield. And these are the same data left and right, colored by either glucose concentration or sodium chloride concentration. And you can see that ideally we want to be on this space over here on the top right, where we have highest titer and highest yield. And what you can see is when we increase glucose concentration, we get higher titers and lower yields. We decrease glucose concentration. But when we increase sodium chloride concentration, we sort of move away, move closer and closer to this point. And this was the most interesting strain that we had, which had both high titer and yield. So to conclude this part of the presentation, we built a semi-automated pipeline for media optimization where we introduced liquid handling, and we could get up to 2 DBDL cycles per week. This was quite effective, increasing titers by 60 to 70% and yield by 350%. We got counterintuitive results where sodium chloride at very high concentration was a very important factor. And also we were able to reproduce things very well between replicates, where we got very high, very small coefficient of variation for all of our controls that we included in every DBDL cycle. And this was also very robust between campaigns, meaning that whatever results you'll get at one point in time or in one lab will translate most likely in a different point in time, and hopefully in a different lab as well. And also, this is very easy to execute with minimal automation equipment, so with only a liquid handler and a biolector. Now, we also went ahead and did more or less the same media optimization for 3HP in Rhodococcus instead of Pseudomonas putida in a number 250. And this was done in a batch mode. And the components that we maximized in this case were DMR total sugars, urea, and magnesium chloride. And the target compound was 3HP. So we're trying to maximize, we're trying to learn 3HP production based on media components. So what we did, we had only 2 DBDL cycles, and we generated 3 components. We selected 3 media designs where we ran on the, 4 media designs that we ran through the Amber. Out of these, only 3 managed to complete. The 4th one foamed up and did not complete the fermentation. And then we trained art only on these 3 conditions that complete the fermentation. And then we queried art to give us media designs that we will run in a second campaign with the only goal to see whether we could accurately predict variable designs and we could predict 3 HP concentration only by training art on these 3 data points. And surprise, we managed to do that. We got a decently good correlation between measured and predicted 3 HP titer, only with these 3 training conditions. And which means that with more DBDL cycles, we can, and even with only 3 conditions, we should be able to tackle even more components than the 3 components that we had. Since we only had 3 components, it's also quite easy to visualize this in a 3D space. What was a bit interesting over here is that, at least what the model predicts right now, is that lower urea and lower magnesium chloride concentrations sort of give the maximum response. And of course, higher sugars give higher 3HP. So to wrap this up, I would like to acknowledge everybody that has worked on this. The initial media optimization paper was a multi-year effort with many people. So we had 18 authors on that paper. And the 3HP is a more recent work that some people from ABPDU have worked on. And contact where you can find us. This is me and the people responsible for leading the 2 parts of the work was Hector from JBEI and James from ABPDU. Thank you.
Thanks, Apostolos. That was a really great tool. So we have a few minutes for questions. Please feel free to put them in the chat. I guess first question for me would be, what's next? So where do you want to take this this strategy?
So what's next? For one, I would like to test this out in ideally either higher scales or with, depending on the budget that different projects have, with more to see where how much can we predict on the Amber at least, how many components we can optimize and sort of try and maximize this. Also, flaviolin is not necessarily very useful chemical itself. So I would like to see whether this can be implemented in more useful chemicals and also in higher scales than the Amber.
I guess good segue then to Deepti's question. So are industry collaborations on your radar here? And if so, how should companies think about working with you?
I think that would be a better question for Hector, but I'm pretty sure we can make this happen. We are part of the ABF as well, so there's definitely the approach to getting a CRADA up and running for that. But otherwise, just feel free to send me an email and we can talk. All of this work is open source, so anyone who wants can get access to the pipeline. But if you want to use art, you would have to license that through LBNL.
Thanks. Yeah, so for those who don't know, ABF is the Agile Biofoundry. It's a Department of Energy group dedicated to more open source tools and development of strains and protocols to improve biomanufacturing. One more quick question here. What would be the limiting factors for the model in the future? Would it be the robustness of the machine learning models, quantity and quality of the data, or other constraints?
So the general thing about this is that this is contingent to having reproducible and low noise data. The better this is, the fewer experiments you need. So I think that without having this tested, and this is also another way that I would like to test this, is to also have process parameters as part of the input space rather than only media designs, which kind of goes away from the media optimization part and goes into the process optimization. But the limitations, as long as you can measure something, what you want to do, and you can finally control what you want to optimize, then practically there are no limitations as long as you can get reproducible data. So these are the three things: find control of the parameters that you're trying to optimize, and accurate and reproducible measurements for what you want to maximize.
Thanks for that. Let's move on to our next speaker. Our next speaker is Jared DeCoste from Prism Immersive, and he'll be speaking about workforce development in biomanufacturing.
Awesome. Thanks, Eric. I'm assuming, are my slides up on the screen?
Yep, looks great.
All right, awesome. Thanks for the opportunity to talk to this crowd today. This is going to be a much different talk than hearing all of these technical talks, which I love hearing and understanding exactly how we fit into this workspace. But I'm going to be talking about next generation technology for training in the area of biomanufacturing and bioproduction. So as many people who work in this field probably realize, right, this field is growing at such an alarming rate, but the workforce supply is something that's going to struggle to keep up with this. All the companies across the world, and especially across the US, are really, really resource limited, and they're looking at training their workforce in person with expensive, time-intensive instruction. Community colleges across the country are looking at how do we train people for the area of biomanufacturing without having the proper equipment for doing this. And at the same time, there's lots of organizations trying to standardize training all at once. There's so many directives across the country right now, especially coming from the Department of Defense, Department of Energy, a lot of other national organizations that have addressed this as a major problem, including President Biden's executive order back in 2022, which said that we need to train a diverse and skilled workforce. The DIPSI awards that came out earlier this year that say that we're trying to bolster the domestic supply, but this also addresses workforce as a major need. And Biomade and Nimble and all the other manufacturing institutes have also identified this as a major, major gap. And why is this a gap? There's two traditional ways to do training. There's hands-on training, which is fantastic, and the way that's actually where you get the most out of it. But this requires such a large capital investment, is very time intensive, and it's not scalable. If you're lucky, you can train at a 1-to-1 or maybe a 1-to-2 ratio. There's online programs that are out there. These don't give you any hands-on training, but they are scalable. But it's also really difficult to assess whether anyone's getting information out of these programs. You can do traditional tests, multiple choice tests, things along those lines. But it doesn't matter if someone gets an 80 or 90%. That doesn't mean that they've actually had the transfer of skills to be in the workforce. At Prism Immersive, we're taking a much different approach to this. And we're utilizing mixed reality. And we want to assess the ability and the promise of virtual and mixed reality to do hands-on bioprocessing, biomanufacturing training, while also mixing it in with the benefits of what you traditionally get in the classroom. We have the ability to use virtual instruction, audio cues, changing of different places in the lab, and utilizing all the promises that virtual and mixed reality have and apply them to this emerging field. And I know I don't have to convince anyone in this room or this virtual room about the large expansion of this field and how many people don't actually get the really good hands-on training or the skills needed to work in a lab on day one. So our core team that is working on this includes myself in the middle. I'm the Chief Executive Officer of Prism Immersive. My background is in science. I'm the scientist of the three. I've worked in the field of biomanufacturing for materials for about the last 7 years, and started as a materials scientist about 15 years ago. My chief operations officer, Dan Beaupre, comes from the educational realm. He worked at National Geographic and was their vice president of experiences for years. And Tom Schmidt heads up our creative division and came from the TV production world and specifically toward applying this towards the virtual reality and mixed reality area. And the three of us have worked together with a team across the fields to really figure out how we can best tackle this problem. So I should start with saying we do have a BioMade Award, and the primary folks who are also involved with this include Amherst, the Biosciences Core Skills Institute, Northeastern University, and Harvard Community College. And then our two original companies, were Experience Science, which was where I came from, and Grasp XR, which was where Tom and Dan came from. And we've come together to build BioSuite Virtual. And we wanted to take a really novel approach on how to build and develop a software for the community. And what we came up with was that we wanted to develop an ecosystem. And we've taken a very ecosystem-driven approach for development of content and understanding the best way to implement this type of training, which is a new and emerging training for a developing field. What you might notice in the lower left corner is something that looks a lot like a design, build, test, learn cycle, and that's what we totally use for doing our development of the virtual reality. And we use 22 pilot sites across the country, and these are a mix of community colleges, universities, industry partners that are really informing the builds as we're going, not just from the perspective of making sure that the content is correct, but also making sure that we're addressing the correct learning design, and giving us actually feedback directly from students. So that's one of the really important points of this is when we're developing this, while we're under development, we're getting feedback from our end user right from the beginning. What you're seeing in the lower right corner is actually one of our test kitchen sessions. So we build new builds, we send them out to our ecosystem. We get on these big Zoom meetings and we basically tell everyone to rip apart what we've done. Why do we do this? Not because we like to hear the bad things, but of course because we want to build the best possible end product at the end of the day.
Carefully pick up the head plate from its holder and then slowly lower it into the vessel.
So what you're seeing here is you're actually seeing a live in-action experience, obviously on a 2D screen, but this is what the user sees inside the 3 dimensions. You're seeing actually our ghost animations on the right demonstrating how to put the head plate into the bioreactor vessel itself, and then the user doing it.
Create a good seal between the head plate and the vessel. On the bottom side of our head plate is a gasket, which will compress against the rim of the glass vessel.
And then along with that, you actually get
our
feedback.
fermentation run. Now,
you get animations telling you what to do
into the highlighted hole and turn it once.
And actually giving you real instruction on why you're doing the types of motions and things that are going on. And this is really important for the user to understand not just what to do, but why they're doing it. We mix this a lot with animations and what you would traditionally see in a classroom. For here on the wall right now, you're seeing the condenser and how the condenser works and what it's actually doing within the bioreactor. There's obviously audio that goes along with it, but I muted it so that I could talk over it. And this actually gives us the ability to have the user better understand not just what to do and the technician what to do, but why they're doing it. And then we mix this with hands-on gamified knowledge assessments. So as opposed to traditional multiple choice questions, fill in the blank, written tests, we try to make our understanding and assessment much more interactive. Ultimately, what we're going to be working toward is not just having these in-experience assessments, but also in-experience credentialing. So we're working with the Biosciences Core Skills Institute to actually understand, to actually develop in-experience credentialing that will be recognized by industry once we're done with this. So these are the phases of development of BioSuite Virtual. Right now, we're still in the first phase, which I know a question will be, well, when do you guys plan on delivering the first part of BioSuite Virtual? We believe we should have the minimal viable product ready by next summer, and we're really excited about that. Moving quickly into building scenario-based learning, basically what happens when there's major safety issues, when things go wrong. How do I address a foaming event, right? How do we address things that go wrong within a bioreactor run? And then moving on to downstream processing, and then eventually into custom processes. So working with industry who have an exact run that they want to do, building that within BioSuite Virtual and helping with tech transfer and things along those lines. We're also excited to move into pilot scale. We're working toward that. Like I said, downstream processing. We want to also go into GMP, pharmaceutical compliance training as well. And then work towards integration with AI to train and develop those custom processes we talked about. With that, I want to take the time. I want to thank Deepti and team for giving me this opportunity to speak. Obviously, Biomade for our funding, our core partners at Amherst, Northeastern, Harvard Community College, and BCSI, the 20 plus other sites that would take me forever to list out. And then I have my contact information there on the bottom, the best place to, I think, see what we're doing, get updates. We do post pretty regularly up on LinkedIn, but feel free to reach out to me directly and check out our website. But with that, I have a couple minutes and be happy to take any additional questions.
Thanks, Jared. It's a really exciting project. So we have one question in the chat here. Is Prism's platform limited to training for lab-based roles or I suppose also pilot-based roles? Or is there a plan to also utilize this technology to train operators for full-scale manufacturing facilities?
Yeah, that's what we're really excited to work towards. So the reason why we started with glass scale is we can teach the fundamentals of fermentation there much better than we can at obviously the larger scale. But we see the scalability as the jobs really start to shift towards that technical and manufacturing role to really need a product like this to train those users. So we're really excited for that to be the kind of the next phase of development for sure.
Thanks for that. And I guess another question I have, do you guys have plans to integrate this kind of workforce development training with job searching and matchmaking to help those who are engaged in these courses find opportunities in the space?
So, I mean, that's a fantastic question. I think our ability to help find jobs is not really going to be the area where we're kind of helping out, but working with institutions who do those larger scale trainings is of large interest to us and are included in our pilot sites. For instance, we do work with NC State University and BTech out there. We're excited to partner with a lot of different organizations as this is coming forward. So I think the reality is we kind of want to leave the things that those folks are good at to allow them to do it and allow the things that we're good at to allow us to do it and do that through partnerships for sure.
One more question. Maybe you have it up, but could you adjust the training pace and level based on the participant's background?
Yeah, that's a fantastic question. That's actually one of the reasons why we love this technology for working for the user is it's really a do-it-your-own pace. So the ability for the user to go back and do repeat a lesson, do it multiple times over and over till they get it right, is there and is something that is, I think, really important to the platform itself. We know that different users will come in with different backgrounds. We hear so often with people coming from a change of job, right? Like someone was in a completely different industry and had no background in a STEM field. They're obviously not going to be at the same point as someone who's transferring jobs or, has worked somewhere else in the operations line. So that's a really important piece to us and something that is absolutely tailorable within the experience.
Wonderful. Well, thank you for that, Jared. Our Our next speaker is Ouwei Wang from Pow.bio. Ouwei is going to tell us about new technologies that they're developing for dynamic bioprocess optimization and control.
Okay, I think everything works. I will probably fly through the slides, and because I want to save some time to do a final demo. First, thank you so much, Deepti and Eric, for inviting me. It's really nice speaking here. My name is Ouwei Wang. I'm the CTO and co-founder of Pow.bio. We are a Alameda, California-based company that provide intelligent fermentation services to the growing synthetic biology industry. We typically operate as a CDMO, so we don't typically present as a software company. So if I start mumbling, sorry, it's fine. Just take that as background noise while you're focusing on your lunch. So today I'll talk about integrating machine learning and digital twins for dynamic optimization in biomanufacturing. So this is basically what happens if you give ChatGPT a summary of your deck and ask it for title. And basically you get this like stitch of all the buzzwords together. So apologize for that. So biomanufacturing will disrupt this $30 trillion industries from meat to bio-based medicine to beauty, collagen, chemicals, textile, food, fashion, fuel, and biofuel, all sort of different compounds. However, many of you, well, this is, I do think it's a real possibility and the future is bright. And many of you probably know that in at least this year and the last year, it was like a real nuclear winter for all the synthetic biology companies. It is very difficult to get funding, but I do see startups start closing again, which is good. So how did we really get here? I would say in the past 5-10 years or so, most of the energies and most of the focus are really just focusing on this first half of synthetic biology, which is idea build and string engineering stage. In this area, they basically have all the fancy tools, automation, machine learning, high throughput, screening, and transcriptomic, and all the fancy stuff. The data throughput and data velocity is really high, and the data quality is also very high. And then you move to the kind of the fermentation lab, and it's almost like time travel back to early 20th century. It is like everything is slow, everything's repetitive and serial in nature. Data velocity is extremely low. The fundamental technology, honestly, is not that different compared to like the original penicillin, like World War II penicillin fermentation. So literally a giant bucket, you stir, you aerate, get cells in there, you get a product. And the process development is extremely expensive because you never have enough tank time. All the cells, all the strands that you spend all these millions of dollars to develop does not really achieve its full potential in bioreactor because you don't really have enough resources to really optimize the best conditions and develop the process. Scale up can be very expensive and time consuming. Many startups usually only get one shot, maybe two shots at scale up. If you fail, the whole company pretty much just collapse. Very similar to like in the pharma space in which people do like clinical trials. So this part and in I would say in the past few years, company fails not because they don't have a strain, they don't have a good strain. Very often it's because they cannot produce, they cannot manufacture at with a good cost of good, or they cannot scale up. So our mission is really to enable the future of biomanufacturing and really address all the problems that I mentioned in the previous slide. We have 3 technical pillars. So there's continuous fermentation, which I'm not really going to spend too much time talk about. And instead, I'm going to focus on the 2 softwares that we've been developing. One is machine learning-enabled self-optimizing fermenter, which we call Sophie. And then the other one is a model-enabled self-driving fermenter, which we call Cassie. So with that, I'm gonna go really fast because I want to save some time just so that you get to see the software. Fundamentally, this slide is basically saying that fed batch is very slow. So many of you, if you run bioreactor, run fermentation, you'll know that fed batch, you basically spend a week tendering a tank, and in the end of the day, you get 1 data point. So that's really the titer, that's like overall like cumulative like product that you care, and then you use that to translate to productivity, to yield. Of course, you're getting a lot of time series data, but then most of the time we use that data really just for troubleshooting, other than like meaningful purposes. So the data throughput is extremely low. Versus in the continuous universe, things can go a lot faster because we are running a continuous reactor. We can let the reactor reach a steady state, perturb the reactor, and then let the reactor drift to a new steady state. So with this, we are able to really unlock the data velocity and ensure that the bottleneck is not at data generation. Because if the bottleneck is at data generation, there's no amount of kind of help in the later stage that can increase the throughput of the overall success, just because you're limited at the very beginning. With that, with increased data throughput, what we then can do is basically generate all different sort of tests and give to a machine learning algorithm, multiple different machine learning algorithms, and really identify what is the best optimization algorithm. So for example, here, basically we were passing this response surface map to one of the machine learning algorithm that we liked. You could see that over time is basically taking steps and trying to deconvolute what is really the background of deconvolute and really reconstruct this true response surface map. So you basically could see all the taking steps and the color of the contour changes. So with that, we deployed that into a continuous fermentation process. And in this experiment, basically we were trying to optimize protein production. So more protein is good. It's a food protein process. Only two variables here, just pH and temperature. We can go at higher dimension now, but this is just like some earlier example. And the output is just protein titer. So initially, you could see that over time, and these are just number of conditions. So every dot is a different condition, different pH and temperature. So over time, you can see at first 10 steps, nothing really changed. But as we kind of go further, that the titer basically increased at the later conditions. And if we double click what happened, you can see that in the first few steps, the temperature went down, pH went up. That didn't really cause any titer changes, dramatic titer changes. But as we increase both temperature and pH, titer start to increase. So basically, the algorithm was able to find the right direction and really just racing toward that angle. And then I'm not gonna really go into detail, but if you double click, kind of like what is the mechanism, what is the mass, kind of fermentation theory goes back into what is the logic that increase the titer. It really coming down to product yield, that we were able to use this to optimize the product yield in a continuous fermentation process. And then what could be optimized in this system? So basically anything that you can meaningfully control and accurately control can be used as a input parameter that include pH, temperature, DO, feed rate, growth rate, medium concentration, all sort of different stuff. And then the output can be biomass yield, titer, product yield, productivity. So basically anything that you can measure with some decent precision and with some decent velocity, it can be used as an output. And this algorithm, honestly, although I'm focusing on fermentation today, is not really limited to fermentation. The same pipeline literally can be used for filtration, for separation, for even spray dry, other processes as well. It's basically like a lightweight optimizer that can be used for multiple different things. And then I'm going to model predictive control as the kind of second software. So now we can optimize our process. How do we really use this information, and can we leverage digital twin to ensure that we can generate a robust model to really predict the fermenter runs. So in this system, so basically we call it self-driving fermenter CASI. And fermentation, again, if you run fermentation, you know that fermentation is extremely chaotic in sort of like a physics way. I do believe fermentation is deterministic. So everything, it follows the law of physics. You can model it with math. However, it is extremely chaotic, meaning that a small deviation at early on can lead to drastic changes, like overall systematic changes later. So very often people describe fermentation as like art. It literally like, oh, it bubbles more than yesterday. It's time to add antifoam. Oh, it's a different color. Something's wrong. Like, oh, it's a start is 100 RPM lower than last run, something's wrong. So it's like a highly artisanal. Very often the lab is run almost like a Netflix cooking show, which is very interesting. That's why, yeah, many people call fermentation as black magic instead of like a science. But I feel like that's wrong. Like we really have to ensure that we have to dial down fermentation, really understand fermentation so that we can actually run robust processes. And that's why we built Cassie. Cassie is really a model predictive controller that can be used to see the future outcome, can flag potential future issues, and really suggest and maintain performance so that we can we ensure that we run process robustly. And use cases, why do we need Cassie? So these are like real life examples, like I literally put from like our internal Slack channel. The first thing is that in R&D setting, like hypothesis testing, I wonder if I change my tank ratio, what is gonna happen? If I increase my feed rate, what is gonna happen? It can be pre-run QC modeling. So like, oh, if I'm like, again, changing all sort of different things, parameters of what is going to happen. If I change my script, what is going to happen? So in this case, right, like we did for some reason, we did have to change our control script. This script failed, couldn't subtract integer from datetime class tab. Basically, there's a coding error, and then we wrote a new script. All this, basically, we could solve by Cassie because we can fit the script to Cassie, and Cassie is literally a real tank, but in your computer. So really predict and flag, like, there's a bug, the script wouldn't run, it's gonna really crash everything after 36 hours into the process. This is something we could use. In production mode, right, like health check. So this is like a more manufacturing setting in which you don't like everything's fixed. You just want to have reproducibility, robustness, and reproducibility. Health check, and then in this case, it's really like useful like overall fermentation health check, like troubleshooting checklist, model predictive controller. For example, if something is deviating, like it keep predicting something, but in reality, like it's not really matching Cassie's control, it's probably going to be like, hey, human, something's wrong. Here's a checklist, do this, making sure you have everything right. So in this example, right, the trainer were noticeably noisier than other tank, swapped the feed inlet to a different port, didn't help. And notice the temperature trend is awkward, and found out the condenser was been off the whole time. The condenser wasn't on, and then we've been having evaporation. That's why this system is like weird. So this kind of things are other use cases for Cassie, basically really almost operating as a co-pilot to help human. I only have 3 minutes, so yeah, it can be used for other things again. So a quick demo. Could you let me do this. Could you see my screen, the other screen, Eric? Am I out of presentation now?
I think so. It looks like
Okay.
you're in the demo.
A tab. Okay, good. Just super quickly, this is a Sophie demo. So basically, we can set different parameters, different steps, minimum, maximum, like minimum bound and maximum bound, because you don't want, because very often it'll be like temperature of 3000 is really good. You want to make sure it doesn't do that and really like cook it itself. And then we have to manage tanks. Basically, you want to make sure you have different tanks of different pH, temperature, DO, feed rate, and then you have different results. You can basically in every iteration you enter a new response and come to action, ask it to propose a new parameter. And basically here you control like, should each tank share this data? Should we have like a EFT-based lag, or should we like turn on or turn off the pH control, DO control, feed rate, things like that. Or whether we want to have absolute optimization. So basically just find your best, achieve your best potential, versus you can compare against a control, just like, making sure you're better than the other tank. That tank is just our negative control, things like that. And in the Cassie space, I'll make sure I'll probably just go easy since I don't have a lot of time. So here basically it's like we already have. So basically what you can do is basically you can load the model. You can see model, load model. Here I'm preloaded everything. And here is where we kind of set all the transporter, different like transporters, girls. This is how we set girls. This is how we set production. Basically, everything is distilled to some deterministic function, and this is how we set kind of like dissolved oxygen set point. Like, what is your tank pressure? How does the TFF separate? What is the TFF separation coefficient and things like that? And then from there, you can get like, oh, here the tank grow, and then the glucose get consumed. We could also, I don't have time. Sorry, I should ask for 30 minutes. So now I think about it. But for example, you could also just change temperature. Let's say, oh, temperature desired, like now it's a yeast process, but I really want to run at 37. What's gonna happen? So basically everything that die. But then obviously there could be a lot of sophisticated stuff you could do, and then the glucose just keep going up because our feed is automated. So yeah, so this is pretty much all. Obviously, I'm happy to answer your questions, and our connection information is here on website and contact information. Thank you for your time.
Thanks, Ouwei. So I think in the interest of time, we can keep the questions in the chat, and Ouwei, if you can stay on, you can please feel free to answer them there.
Yeah, happy to.
We'll go ahead and move on to our next speaker. Our next speaker is Andrew Chiappetta. Andrew is with BioReact, and he's going to tell us about all-in-one fermentation software.
Great. Okay. So thank you very much for giving me the opportunity to speak at this expo. A lot of really interesting technologies here. Today, I'm going to be talking about the BioReact platform and what we're doing at BioReact to basically give scientists and engineers, like the ones that are speaking and listening in here, an all-in-one tool for fermentation software or fermentation process optimization and data management. Myself, I'm part of the BioReact team. I'm a chemical engineer, been doing fermentation process development and optimization for about 12 years now. And a lot of the things that BioReact is solving stems from problems that I think a lot of us have in the lab when we deal with managing data and optimizing our studies. So in the lab, scientists don't have a dedicated platform to make sense of our data. We have our bioreactors generating a ton of online data, and we're running quite a bit of experiments. And then we have our offline experiments that are continuously generating massive amounts of experimental data. And this leads to some challenges where we're spending a lot of time manually processing data, moving data from one software to another, copying and pasting data, saving data in different file formats. And I think one of the most frustrating things is aligning data, especially when we look at offline grab samples for OD600, for HPLC. So really what this leads to is errors, many different errors, and just time spent dealing with data. Oftentimes low yields or struggle scaling up because we spend a lot of time just wrestling with this data and essentially not enough time really focusing on running experiments, paying attention to what we need to during scale up, and not spending as much time either in the lab as we could or really managing some of these more technical challenges or process related challenges because we're just being inefficient with our time. So how can we solve this problem with BioReact? Well, when we look at a traditional workflow, I know this is something that I've experienced quite a bit, where in a traditional workflow, we will run our experiments, let's say on the bioreactor, we'll save a lot of our data into OneDrive or Google or something like that. When we're done our fermentation study, we will download the Excel file or the CSV files, open them in Excel, manually align this data. Maybe we'll manually add our OD600 time points into that same Excel file. We'll spend a lot of time plotting things and plotting trend charts and doing some basic statistical analysis, making things look really nice for presentations. And then oftentimes we'll take that data, we'll port it over to another system, for example, R or JMP for this basic statistical analysis. Nothing too fancy, but it still takes time to perform this analysis on the data to demonstrate that our process changes are actually having an effect and that we can see the effect of different set points and things like that. And finally, once we have all of this data analyzed, all these nice trend charts created, then we'll go over to PowerPoint and we'll prepare our nice PowerPoint presentations for our teams to show that we actually know what we're talking about. So that's a lot of different softwares. It's a lot of time managing all these different systems. And of course, we have to spend money on all these different systems. So the goal of BioReact is really to replace this end-to-end workflow. What we want to do is be able to ingest data automatically, perform all of these different functions in one place very easily to save people time, effort, and energy. So what exactly does BioReact do? Well, BioReact transforms fermentation data into actionable insights. So the first thing, data management. So it allows for automatic data parsing and automatic data ingestion from bioreactors and from offline instruments, such as your HPLCs or your spectrophotometers, essentially anything that is able to output data to a digital source. We can grab that data and bring it into BioReact, which just saves quite a bit of time. And I think one of my favorite features is the data alignment. So just based on your time series, as long as you have units for things like your sample time, let's say if it's fermentation duration, you can align all this information very easily into one large data set without having to handle it yourself at all. Then we get over to the visualization, a really great tool for generating things like heat maps, line charts, and performing statistical analysis. The great thing about this is you can overlay multiple different experiments. You can overlay multiple parameters from the same experiment. Very much similar to trend charts that you would want to create yourself. There's full flexibility here. So you can adjust axes, colors, which data is being shown. And for up to 20 experiments, you can overlay this data together. So replaces a lot of the function that we would use Excel for to create these trend charts. And yeah, some people can create them themselves in Python and things like that. But this is an out-of-the-box solution ready to go right away for your scientists and for your team. The next feature is the AI optimization. So with BioReact, we can perform bioreactor parameter optimization. So with at least 10 data sets, we can then go ahead and assist the scientists with determining what the optimal parameter set points should be for their different runs. So whether that be temperature, dissolved oxygen, as long as the data is reasonably designed and there's enough variability within the data to assess the effect on the output. And also we can perform media optimization and design of experiment. So assisting with designing the experiments, assisting with collecting data from these experiments and analyzing the data as well. So this is often what we would use things like JMP for or other DOE type software. And it's all within the BioReact system as well. And finally, equipment integration. One big feature of BioReact is that we can integrate with equipment, automatically pull data, but also assist in controlling this equipment, depending on the level of security required in the lab. You know, some labs may not want to do that, but we can assist in controlling equipment, changing set points to achieve an optimal fermentation process. So essentially, it's a co-pilot for scientists to gather their data and also to control the equipment. Currently, BioReact is trusted by over 170 scientists using this software from companies like Fujifilm Cellular Dynamics, BioVect Innovation, Live and Proteins, and OraBio. And we're proud to announce that this year we are part of the TechStars universe and have also been into the BioMade program and the NVIDIA Inception program. So we're seeing a lot of uptake in the software. We're seeing a lot of people starting to use it and really like using it. We're finding that it's saving people time and effort in analyzing their data and planning their experiments. So this software, I think the biggest power, as I mentioned, is data alignment and management. We can handle online data from all bioreactor sources. I've named a few here from Sartorius, Inforce, Beckman, Eppendorf, etc. But also custom-made pilot and commercial scale reactors. As long as we have a template of what the data looks like being output from the system, we can set this up on your BioReact platform quite quickly and allow you to get up and running in as little as 1 day, just basically ingesting data automatically. And the great thing is offline data is also easily ingested by the platform, whether it's a drag and drop type system from an Excel file, or whether you want to automatically integrate this into your workflow so that it's pulling data automatically from the system. Once again, as long as we know what the data looks like, we can help you integrate it into your workbooks. So things like spectrophotometers, HPLCs, biomass probes, off-gas analyzers, cell viability analyzers, essentially any type of data as long as there's a reasonable structure to it, and we can relate it to a time series or however else you would like to relate it to your fermentation. We can bring it into the workbook and help you analyze it. So I've put a few plots here of just what the system looks like currently. On the left, we have some OD600 plots from about 15 different fermentations. So this is just something very simple, overlaying OD600 from all these different fermentations to observe what we see, the kind of trends are between the different runs. We can also display the information about these runs, if there's any different strains that are used, any different medias that are used. But if we wanted to overlay things like dissolved oxygen curves, we can also do that. And on the right, we have two different trends of a high yielding on the top in blue and a low yielding batch in red to observe what are the critical points in the fermentation that are really causing differences in the output. So this software can more easily and more quickly help you identify areas in your fermentation that are resulting in either high yields or low yields or anything else of interest. Let's say you want to minimize certain byproduct production. That's something that we can also help you do. So it helps really quickly with analysis, and it's quite flexible in terms of what you would like to achieve. The next thing that we can do is DOE designs. So very simple DOE designing. A lot of these DOE tools that we use, sometimes they're very complex, and they don't necessarily need to be complex. DOEs are not something that needs to be super complex to design. So what we can do is decide on the number of parameters that you would like to optimize. And this is totally user dependent. If you want to look at 5 different parameters or even just 2 different parameters, and the ranges, whether you want to look at set points or whether you want to look at range, you can input this into the tool with a simple screen like you have here. And even if you want to look at different medias to help kind of block your experiments or to look at optimizing your medias, you essentially input all of this information in, and what it will suggest is a series of experiments and how you should execute these in different blocks. What you will then do is upload your data into this platform, and then you can more easily analyze the data from these experiments. And when we look at what do the outputs look like of the statistical analysis, we can do things like factor importance and principal component analysis along with different heat maps. So we can determine primarily where is the variability coming from in your experiment. So if you want to understand what is driving variability in my output, which parameters are the most responsible for that. In addition, we can understand or help the user understand what is driving yields. So if you want to maximize yields, which parameters are primarily responsible for driving high yield or low yield in your batch? Or like I mentioned, if you want to look at host cell protein or any other factors that you want to minimize or maximize, this is something that we can help users understand very simply with a few clicks. And we also offer AI-driven parameter and media optimization. So as I mentioned, with as little as 10 batches, we can use our custom-made AI algorithm that's trained on over 7 million data points to very easily understand how to basically perform a more optimized process than what you're doing right now. So essentially what it would look like is you upload all your data, and based on the data that is uploaded and the model that has been trained on similar type organisms, the platform will output parameter set points. For example, here we have dissolved oxygen in terms of how you should be running your fermentation. So this can be either just a set point, this could be a series of set points, or this could be a profile with an equation, depending on what your input data looks like and what an optimal set point would be. You can then you can take this equation or these set points, plug them into your fermentation system and execute a run, then go re-upload this data for more efficient optimization with fewer iterations. So overall, this is just going to save scientists and engineers time, make things a lot easier to optimize, and just spend a lot less time just struggling with this data. So that's all I really want to talk about today. Show you a little bit about the platform. Now we have free licenses available at app.bioreact.co. I encourage people to go take a look. You can get a free license. You can download some data sets. You can play around with uploading the data sets, see how easy it is to drag and drop Excel files, drag and drop offline data files, and visualize and optimize data sets. So really convenient, really easy. and I think it will save people a lot of time and effort. And if you'd like more information, you can reach out to sales@bioreact.co. And I'd just like to thank the organizers for giving us time to speak today, and I really appreciate it. Thank you very much.
Thanks, Andrew. That's a really great technology platform here. So maybe one quick question for me. One thing we deal with quite a bit as a scale-up facility is tech transfer. And when different groups have different data management platforms, it can sometimes be more challenging for us to access their data or their protocol. So I'm wondering if you could speak to how your software manages that.
You know, that's a great question. That's something that we get quite often. The great thing is that we have some CDMO and CMO customers that what they will do is they will give access to the BioReact platform to their customers, for example, and their customers can upload this data, or vice versa. The customer can give access to the BioReact platform to the CDMO. And what it allows primarily is looking at all the data, very easily sharing data without having to send over files or give access to SharePoints or things like that. You add them to your project, they can see all of your data or limited data that you would like to share with them. And you can also demonstrate what an optimal process would look like in terms of set points. So it just makes sharing of this data very easy. It makes understanding scale up very easy because you can do things like input KLA parameters, tip speed, volumetric airflow rates, to more easily facilitate the scale up. But if you already know exactly how you want to run it scale up and you're doing things like sharing parameters, you can just give them access and share this data very efficiently. So it just facilitates the transfer just much more easily.
Hey, thanks for that. And if anyone
Thank
has
you.
any additional questions, please feel free to drop them in the chat. Our next speaker is Alex Felt from Invert and Inscripta. And Alex will be telling us about a interesting technology platform they have to smooth scale up of biomanufacturing.
Hi, everyone. My name is Alex, and I lead business development at Invert. Today, I'm joined by Sylvie from Inscripta, and we'll be taking you through a bit of a case study on our work together and how Inscripta was able to speed up some of their fermentation process development efforts, as well as scale up using Invert. So with that, Sylvie, I will pass it to you.
Hi guys, I'm Sylvie. Currently a process engineer at Inscripta. I work on the fermentation team with the Ambers and Sartorius 2 liters, and I want to introduce Inscripta as well as its core technology and give you an overview on how we approach fermentation. So I'm sure most of the audience are aware of like the biomanufacturing opportunity to produce molecules that are not possible using traditional chemical processes and create scalable and sustainable supply chains. The challenges in our industry are long timelines for strain and process development with the high R&D costs. And I'm sure you heard the phrase, the valley of death, which refers to the R&D results not exactly predicting performance at manufacturing. Inscripta is able to solve these challenges by utilizing its core technology, GenoScaler, which is the world's first whole genome engineering platform. We use minimal strain design and predictive models for robust scale-up, which enables us to bring products to market with short timelines and reduce costs. Next slide. Inscripta's technology enables us to build strains that are designed for manufacturing, and common industry practices are heavily engineered microbes that are unfit and genetically unstable, resulting in terrifying scale-ups. GenoScaler technology allows us to utilize precise gene editing, resulting in minimal design and fit strains. Inscripta's microbe has high fitness and productivity, as well as stable genetics, which makes scale-up a success. Next slide. Here is our DPTL cycle. It starts with a high throughput screening, or HTS, where thousands of genotypes are screened using predictive plate models and high content imaging. Selected hits from our HTS are screened in 250 ml bioreactor platforms. We have the Amber, which is our in-house, and we do outsource some to Culture Biosciences. Process development is performed at numerous scales and sites ranging from 250 ml to 300 K liters. And scale-up activities happen at the CDMO site to support our different phases of the project, ranging from sample generation, scale-up, and manufacturing. Next slide. So So as you can see here, there are a lot of data from different scales and platforms that are not structured in the same way. Utilizing common data visualization tools such as JMP or JUMP or Excel to join, clean up, and visualize data across scales becomes very cumbersome and time-consuming. Also, if you want to do any machine learning, you're going to have to get your data structured properly. Next slide. So we want to use the best practices for developing and scaling fermentation processes because there are many challenges with this. With R&D, the cycling times are important so we can keep the company moving forward and operating as a lean team. A solution is needed to quickly turn around data, and this is where Invert comes in.
Great, thanks, Sylvie. So I will skip the problem statement because I think Andrew from BioReact did a really nice job covering that. But just to quickly introduce Invert and who we are and what we do. So we're a cloud-based software company or cloud-based platform. The scientists and engineers use their hub for bioprocess data. We've been around for about 3 years, and we're on a mission to bring modern software to bioprocess development, scale up, and ultimately control. So what does that look like? It's a single place for bioprocess scientists, engineers, tech transfer teams to effectively do all of their work. So teams like Sylvie's over at Inscripta. And the way this looks is we can connect directly to equipment. So all the different bioreactor types, as well as ELNs or any sort of existing databases. Invert automatically takes that data coming from all these various sources, harmonizes it, so normalizes it to, say, your time zero. It also is able to parse all of those results and effectively store it on a run basis. And one of the ways we're able to do this as well is we're entirely unit aware. So any sort of measurement that comes in has an associated unit. That means that calculations can be performed, formulas can be created, and everything is effectively synced so that you can compare across scales. And with that, within the platform as well, you can also analyze all your results as you would in JUMP or in Excel, as well as model bioprocesses. You can see a nice little screenshot of what that looks like there on the right. So taking a look at Inscripta's overall data workflow, I would call it. So going from the bottom, which is source, all the way through data processing, storage, and analysis. As Sylvie mentioned, they generate data across many different scales and sites, right? So that's the Amber, which is run in-house. That's using Culture Biosciences, which is a high throughput, small scale platform. And that's with the various external partners at the larger scale, so CDMOs and CMOs. The sources of this data and the formats that they're in can vary quite widely, as well as how clean or messy they are, right? So CSVs, Excel sheets, PDFs. The Inscripta team previously was taking all of that through tools like JUMP, Excel, performing their QC, maybe putting some into their electronic lab notebook, which is where a lot of metadata and sample results are stored, that then can be passed into a SQL database and then back into JUMP for analysis. Final results and outcomes, maybe as reports or images, were then stored in Box. So as you can see, pretty manual, very, very typical. So this is what I see from pretty much all of our customers, both in SynBio and in biopharma. So nothing atypical here, but I think that's unfortunate and it's something that we're looking to really change. After Invert, so the two of us, our two companies have been working together for just about a year now. This is what the data workflow looks like today. So same sources, ranging from the Amber to Culture to CDMO and CMO. All of that is automatically brought in through Invert in automated data pipelines. So the Amber, we connect directly. That is live coming off of the system. So the second a run goes up, all of that data is being fed directly from the Amber into Invert for analysis or monitoring. The data from Culture, CDMOs, and CMOs, that ranges from Excel to PDF. We have quite a bit of technology that we've built internally to be able to parse those. So the format of those sources can vary, and they can be fairly complex, right? So it's not atypical for a batch record to have 12 tabs, a lot of sort of human readable content, but less machine readable. We can parse all that automatically and bring it into Invert. Data from their ELN can also be brought in via the API. So a lot of systems out there have APIs that we can access and automate data input into Invert. We can also push data out via our own API. So that goes into Inscripta's database for future sort of storage or posterity. And then with Invert, they can perform all their analysis, any sort of calculations. So I mentioned that we're fully unit aware. It means you can do molar conversions, calculate things like growth rate, KPIs, like yield or productivity. And all of that can be done automatically within Invert itself. And then reports are also able to be generated. So documenting your experimental outcome, transferring that to an external partner, or even internally for review. So what does all that mean? What was the impact of that? So through the use of Invert and everything I just mentioned, Inscripta was able to really optimize that development cycle, so that DBTL cycle, to run more experiments. And then they actually successfully scaled to past 1,000 liters while keeping that same lean team. Some more details there. So by eliminating all these manual data processes and improving that productivity of the lean team, they saved over 40 hours per month of data processing time. With that saved time, they were actually able to also reduce turnaround time between experiments. So we see quite often, and I'm formerly a process engineer, so I had to deal with this quite a bit. On Fridays, maybe when experiments come down, you're spending the entire day both in the lab taking that experiment down, but also gathering all your data, trying to figure out what the results look like, and then design the experiment for the next week. That's really hard to do all at once, especially with a smaller team. So in eliminating all those manual data workflows, Inscripta was able to actually cut that turnaround time from a week to a day, and then run an additional 2 experiments per month with the same overall capacity. Finally, because all the data is being funneled in through Invert, and that's both small scale and CMO, they're able to actually compare those results more easily and then be able to design experiments both at the small scale and with their scale-up partners to find better outcomes there. On the right here, you can see some of the results from Invert as well. Great. Now looking forward, we're set on time here. We are in 2025, Inscripta and Invert are actually partnering together to deploy some machine learning models for both new and existing programs. So within Invert, out of the box, we have a variety of models that users can use on their own structured data within the system. We also have notebooks, which is a free-form Python notebook that data scientists typically like to use to further explore their data or create their own models. In addition to what's existing within Invert, we also offer modeling services. So Inscripta and Invert will be partnering this coming year to develop novel machine learning models, so things like hybrid modeling and some other approaches as well, to further optimize yield, productivity, as well as be able to predict scale-up performance and really de-risk that path. So with that, here's our contact info, and that's it. Thank you very much.
Thanks, Alex and Sylvie. Really interesting. A quick question for you about metadata and observations. So I know a lot of times when we're looking at this kind of data analysis, we get hung up on one-off events or mechanical failures or qualitative observations that aren't easily tracked in the data. I'm wondering if you could just speak to how you guys manage that in your software platform.
Sure, absolutely. We have a few different ways of doing that with an Invert. So one, you can log events. So that includes pictures, that includes commentary. So that's actually tied to the actual run itself at a certain time. So you can say at 24 hours for this run, there was a discoloration that happened or there was a pump failure. That actually can also be used as metadata. So if it's tagged appropriately, it can be used to model out rate of failure and be able to identify deviations in their root causes. So we really try to make it as easy as possible to tag events and log the associated data with those events such that they can be easily looked upon at a more larger level.
Our next speaker is Yufei Sun from Washington University in St. Louis. Yufei is going to tell us about a really interesting project to make the fermentation and bioprocess literature more machine readable and available for larger scale analysis. So, Yufei, you can go ahead and take it away.
Hi, thank you, Eric. Just gonna share my screen real quick. Yeah. If everyone's seeing this okay, I'm Yufei from Washington University in St. Louis. I'm a third-year PhD student in the Tongue Lab in the Department of Energy, Environmental, and Chemical Engineering. Today I'm going to talk about a project that we're going on. The name is the Industrial Microbiology Publication and AI Crowdsource Toolbox, aka ImpactDB. So the concept is that because synthetic biology have a very complex influence factors, researchers usually try to optimize the titer, yield, and rate for fermentation processes. But there are so many different factors that have influences on these factors that we need to optimize. Therefore, using tools like machine learning or artificial intelligence or genomic skill models, etc., will largely help this process to reduce costs and improve efficiencies. But however, training these models usually requires a high quality and a large amount of data in order to obtain a model that are accurate. But within a single lab, the data generated is limited. And if you check literatures, data will be spread across many publications and formats. So there's a need for a structured database of synthetic biology data. So here we introduce ImpactDB, the Industrial Microbiology Publication and AI Crowdsourced Toolbox, originally developed by our former lab mate Garrett Rowe, who is currently at an assistant professor at the University of Hawaii. So currently, our database has around 9 species, and it's continued to expanding. Each paper will be structured under a specific species, and results will be associated with specific papers. Here is a typical example of a listed paper on our website. So this paper on the left side will have the publication date and the journal name, followed by the article title. The article title will also serve as a link to the original DOI of the publication itself. We also listed the abstract because it's open source. And then we list the author names and all the fermentation results that we extracted from the paper. So here is a typical example of how our fermentation results will be structured. We'll have the titer of the specific product. We'll have almost all the bioprocess condition data, and then we'll have another section of genetic background of the strain. So on the left side, we'll have the substrate concentration, the time, oxygen levels, almost all the factors that you looking to optimize for reactor condition. And then we will also list what modifications to the genes, to the strains are listed. So currently we have 2 different ways to continue to grow our database. The first one is by manual reading, crowdsourcing from different labs. So this involves researchers manually read through articles and then extract the fermentation data that we wanted and put it in our standardized format. We've been doing this for almost 3 years now, and we've got thousands of results, and we've added almost 1,900 papers on our website. But we all know that manually reading through publications is not the best thing to do or the most interesting thing to do. Since we have AI tools like ChatGPT developed recently, we want to use a second way of AI accelerated, followed by human supervisions, to accelerate this data extraction. Currently, our database also have some other toolboxes for researchers to use, like molecular inventory, which listed, I'll talk about later, and then also a pathway map and potential feature like knowledge networks. We also have design toolbox to help researchers to predict titer rate and yield. And then researchers are also able to download our database for the training of their own machine learning models or other potential uses. So here is an example tool of titer prediction developed by also my former lab mate Jeff. This is a Ouwei-based titer prediction tool, which you can enter the type of product, your substrate, and then concentration, and our other conditions, and it will give you a titer prediction, which we can achieve 80% plus accuracies. We also have a pathway map and molecular inventory, which is mainly designed for strain designs. Is a quick reference for researchers to use when they're trying to produce a certain product. And the molecular inventory is mainly to assist machine learning models, which consists of some chemical and physical properties of the common substrates and products, including energy of formation, boiling points, etc., which sometimes you'll need for genomic skill models. So now I'm going to talk about our AI-assisted workflow. Essentially, we use we input the scientific articles, and then we use through some prompt engineering, we would extract the information from the paper into a standardized format, and then we bulk upload it to our website. The important note is that because large language models are not 100% accurate, we still enforce human supervisions in these following steps. So here is an example of large language model extracted data that produces used to be trained for a machine learning model that predicted titer for publications. So lastly, I want to talk about our potential work in the future. So we are planned to also use the extract information and with tools like GraphRag to produce knowledge networks, which can better help researchers to guide through their strain designing or other processes, which makes our database more like a one-step tool for fermentation scientists to use. And lastly, I would like to thank the NSF for funding this project, my PI, Dr. Yinjie Tang, and all the Journal Club members, and also the founder of ImpactDB, Dr. Garrett Rowe. And I would like to answer any questions if.
Thanks, Yufei.
Mm-hmm.
So we have time for a few questions here. I guess one to start you off. So we're in the business of both consuming and producing published studies. So I think having gone through the process of taking the existing literature to pull the data out, do you have any recommendations for how we could be better at publishing new studies to make that process more painless for future researchers, or to feed into your process more directly?
That's actually a great question. So I know most people's concern will be since you're pulling off data from previous publications, there might not be much novelty into it. But we do see in cases where a specific advance or say species A, we're studied to produce a certain product that we're achieving higher productions, we should be able to use this as a template to other species. This is just like an example, which can help researchers to open their mind or try new stuff to actually produce some novel findings. And also, the optimization, the data that we use to train the machine learning models can actually give you some predictions and recommendations during strain designs. Traditionally, some genes, researchers might have the knowledge of knocking out or modifying specific genes to increase the production titer, but sometimes the machine learning model will actually guide you to do a different strategy that will actually further improve the production.
Great. Have you guys thought about maybe working with the journals more directly to make the manuscripts more machine readable, or reduce the burden on having to scrape the data?
Yeah, so this it's very hard to do because most of these publications we have to actually manually extract the information from them, and most of the journals doesn't really offer APIs for us to actually obtain data from them. So I think that's why we're doing this, to actually centralize the data for provide easier access for researchers or scientists.
Yeah, interesting. It's a tricky problem, I guess, to get all of that historical data properly wrangled. Have you thought about using this kind of thing as a training opportunity? I know we've talked about workforce development here, but for students in the field to look to do some of the data management and use that to gain a better feel for how fermentation studies are structured and how the data is structured and how they can pull out relevant information from publications.
That I'm not 100% sure. We've because previously we do manual extractions, and we sort of use this as also as an opportunity to train younger students who have less background in biology field to actually locate the information or get used to the knowledge that we're interested in. But we haven't thought about training workforces in this format, I guess.
Oh, great. Yeah, thanks, Yufei. So if anyone has further questions, please drop them in the chat. And I'll hand it off to our next speaker, Tom Trerynor from R2DIO. And Tom is going to tell us about strategies for maintaining high data quality and data management in bioprocess scale-up.
Great. Thanks for the introduction and the opportunity to speak today. Our company is R2DIO, and with our radio tower logo, or radio dish logo. We also call ourselves Radio. And I'm going to talk to you about how radio software and services are unlocking new ways to make things better, faster, cheaper, and together. I thought I'd start by highlighting this article. Maybe some of you have seen it already. If you haven't, you should definitely check it out. It is written by a couple of my favorite people in industrial biotechnology, Jason Crater and Jeff Liebensee. And they have distilled their incredible experience in the industry into just 3 bullet points. 1, begin with the end in mind. And 2, be diligent in the details. 3, prepare for the unexpected. This is a great read. And I thought it was also a great way to introduce you to radio and our software and services. Let's take their advice. We'll begin with the end in mind. I'll tell you what you'll be able to take away from the rest of my presentation today. Guess what? We're going to help you be diligent in the details. Our software is a new kind of category, call it a process data fabric. It's truly the missing piece. It's the thing that's going to weave your current team, your current instruments, your current tools into this engine of ever-improving traceability, analysis, and insight. We are going to help you be diligent in the details. And we're going to help you prepare for the unexpected. Radio from day one has been designed to help you do this. Our dashboards, our data warehouse, and our dynamically curated relationships, they complement your favorite data analysis and visualization tools, JUMP, Python, with the power to detect anomalies, truly accelerate your learning from today all the way through to commercialization. I want to highlight that we're really excited to announce that we'll be launching the free self-registering version of our product in February. If this is something you'd like to try, you can write us anytime today or later at waitlist@radio.com to learn more. Also want to announce that UC Berkeley and Radio are submitting a white paper to Biomade's currently open project call. We're looking for 10 partner organizations to join us. And as I will show you in some of the slides today, we will, for no cost to your own company, we will help you make your bioproduct assays and the teams that rely on them at least 4 times more productive. Again, if you'd like to learn more about this, you can write us at 4x@radio.com. So again, we will begin with the end in mind. I'm going to work backwards from some customer testimonials through to some of these case studies, like the kinds of impact that our Biomade teaming companies will be able to get access to. And then we'll make a U-turn and we'll start heading back towards the end of the presentation finally. Really pleased to be working with a company many of you know in San Leandro called New Culture. They're using precision fermentation to make dairy protein, and really excited how they were able to use radio to build initial tooling for everything from their biology team to their fermentation, their DSP, their food formulation, their sensory testing. And they were able to do this at multiple scales, and they were able to do it all in just 3 months. They told us that they had investigated 15 different tools before down selecting to 5 and then ultimately choosing radio. Really happy with their experience using the platform, and encourage you to talk to them if you know them. Also working with a company called the Protein Brewery, another food tech company, precision fermentation for sustainable protein. They have been using radio for 3 years now. A really interesting fact is that they have no software team, no data science team, no IT team. They were able to use radio to set up all of their own bespoke digital tooling, integrate all of their operations, again, the biology, the fermentation, the DSP, the food formulation, the sensory testing, get on all of it onto the platform. We asked them recently how much time they spend maintaining that end-to-end digital tooling on radio. They told us about 2 hours a week. So this was the problem that felt like it needed to be solved, was how to bring all of these incredibly different teams with their remarkably different ways of collecting data and analyzing data and bring it all together onto one platform to figuratively and literally put everybody on the same page. They are doing that with radio. Love the fact that they describe using our product as fun and not simply user-friendly, but they say that too. And importantly, they had experience using other platforms at the companies they'd worked for previously, but they felt like the data was, very painful just to put it in there, and then it didn't come out again. What's the point of that? So with radio, their data is actually getting used. This third example predates radio, but it really helps to illustrate how radio's approach can unlock these new ways to improve even very mature biomanufacturing processes. So when I was previously at Zymergen, we worked with this Fortune 500 ingredients company. And again, this radio approach, when you make it possible you make it possible for the contributors, the scientists and the engineers to basically build their own data infrastructure, to do it fast, then they can build out these multi-level dashboards. They can curate all the relationships in their data, make them mineable for insights. What we achieved was the ability to predict at scale fermentation KPIs with less than 3% error. In fact, I'll show this to you on the next slide. This system enabled our team at Zymergen to find multiple strains that improved over their manufacturing strain, despite the fact that they had spent years trying to achieve what we were able to achieve in just a few months. Here is the data. You can find it yourself in a patent that is very public. And there are over 100 strains on this chart. They involve perturbations to central metabolism, secondary metabolism, on pathway, off pathway, doesn't matter. This batch fermentation process that we operated in microtiter plates with a throughput of 10,000 strains per week was capable of predicting fed batch, stirred tank, DO, pH-controlled bioreactors with a prediction error that was just 3% of what was observed in the tanks. Most people still think this isn't even possible, but we've done it. It's been published in the patent literature. And again, radio is really founded to make this approach to collaborative R&D just much more accessible and impactful in our small but very promising and growing industry. So when you are able to do this, right, you start with this initial condition that's so frustrating and so familiar to many of us, right, where there's just this low, weak correlation between what we're able to measure in these convenient, cheap, scalable, batch fermentations and what we need to achieve in a more manufacturing relevant system. And when you deploy these data systems and these feedback loops, you really tap into all the expertise in your team and you enable them to accelerate this improvement of the measurement system to the point where, in this case, those prediction errors less than 3%. And so what this meant was, we had some strain engineers, all of their hard work, all of their, carefully designed strain ideas were tested effectively, right? Reduced false negative rates, reduced false positive rates. Fewer of those truly improved strains just get lost in the noise. By the way, noise, it doesn't behave like a normal distribution until you make it behave like a normal distribution. We'll talk more about that in a minute. And so it's important to highlight that, like, we couldn't have done it without the subject matter expertise of our team and these data systems that basically enabled those scientists and engineers to drive the continuous improvement of their data collection and analysis, in addition to harnessing their training. So I thought I want to take you on another case study, one that's very relevant to biomanufacturing, even at scale, but of course also to what happens in the lab. This one's from Amyris. And here we'd been operating this, call it the old bioproduct assay, for years. This was a fully automated process. But what this illustration of the controls, the standard materials that were running through our analytical method, what this chart, called a control chart, is illustrating is that even this fully automated method, the noise is simply not distributed like a random normal curve. We have outliers, we have clusters and trends, we have this weird upward trend that's kind of happening over and over and over again, right? So even more important than the automation then is the feedback loop. It's the ability to put the data in front of the subject matter expert as soon as it's available so that they can act on the signals in the data and really accelerate the learning process and even the quality of the data itself. So in this project at Amyris, we implemented this new bioproduct assay. It took us about 3 months to kind of work out all the details, but only 3 months. And as soon as we implemented it, you can see the difference was night and day. This new method used the same instruments, the same people. It was a more data-intensive method of measurement that required writing down more numbers, crunching more numbers, but without the kind of expensive six-figure investments in new equipment. These teams were able to substantially improve the quality of their analytics data. That's not all it improved. As soon as we implemented these new methods, the reproducibility of our stirred tank fermentations, something that we were running 60 times a week, right? I mean, we're talking, six-figure burn just to operate these fermenters every week. Immediately, the new bioproduct assay reduced or improved the control of the fermentation process from just 4%. Well, it was already low at about 4%. Now it's only 1.6%. That may sound like unnecessary. And if you think that, you're wrong. Because what happened is we've been operating with 4% for like a year before this project. And our fermentation team of nearly 20 people was, doing these multi-factor DOEs, putting the data into their statistical software time and again. Software was replying to them saying, your ideas don't matter. Your p-values are greater than 0.05. Move on to the next experiment. Throw all these ideas out. And that's actually not what the statistical software should have been saying or what we should have been hearing. What we should have been hearing is, look, these are good ideas, but your process has matured to the point where the signals associated with those ideas are indistinguishable from noise. And what's important to realize is that doesn't mean the signals don't matter. The noise itself, 4%, is actually so large, it is obscuring $1 million improvements. No joke, right? So as soon as we implemented this new bioproduct assay, a single fermentation run was able to generate a quality of data insight where previously we would have had to run 6 replicates. And now, of course, we didn't run 6 replicates. We kept moving on to the next idea, and we had this very high false negative rate as a result. So again, as soon as we implemented the new methods, we had the same team with the same equipment. Suddenly, all of these DOEs are generating p-values less than 0.05. The improvements that we learned to improve yield and productivity in tanks, they all translated to our 1 million liter scale facility in Brazil. It was a real thrill to be part of this project. And again, like, why did this work? Because we were able to really tap into the expertise of our analytical chemists, our fermentation engineers, our firm techs, and we were able to do it by basically deploying these data systems that put the information at their fingertips as soon as it's available. Joel Cherry was our president of R&D during this period from 2008 to 2019. And when I saw him just a couple years ago, he said, look, Tom, like this work that you and our team did to improve data quality, it truly reduced our product time to market by 50%. So talk about beginning with the end in mind, right? Wouldn't this be a great thing for your own companies? This is why we founded Radio, right? We really want to make this accelerated bioproduct development trajectory accessible to everyone. The problem, the challenge for our industry is most of the time you spend in bioproduct development just does not look like the earliest phases of bioproduct development. That early period of time where we win our pre-seed and seed and Series A funding is a period of time where it's incredibly thrilling, right? We have these small teams, and yet we're making really fast progress with our technology development, our strain engineering, you name it. And then, well, inevitably, that low-hanging fruit is going to be in the rear view mirror. And so we spend most of our time trying to scale and commercialize our bioproducts. On this other part of the S-curve. And the reason why is because at this point, most of the signals that matter to the bottom line, those $1 million signals even, they're indistinguishable from noise unless we do what I've been describing here, which is really build data systems and commit resources to constantly measuring the variation in our processes and empowering our colleagues to act on those signals and drive continuous improvement in the work they do. This is why we built Radio. And this is actually very relevant to this BioMade teaming opportunity that I mentioned. Again, UC Berkeley and Radio, we're going to be writing up a white paper for the currently open project call. We want to help 10 partner organizations basically do this, make their bioproduct assays. Basically, all the people who are operating those bioproduct assays, make all the fermentation engineers, DSP engineers who are relying on those bioproduct assays, like literally 4 times more productive at least. Again, we think we can do this without any cost to your companies. If this sounds interesting, please write us 4x@radio.com. Our plan is for a funded project, assuming it's funded. It will pay for your access to the software, for tablets, barcode scanners, other accessories that help you to really operate these best practices with a minimum of disruption, no new hires, no downtime, nothing slowed in your labs. Doesn't matter what kind of fermentation or DSP equipment you have or analytical methods. we want to work with you. So just write us, let us know if you're interested. Yeah, so Radio's process data fabric, really think of it as the missing piece here to unlock these kinds of capabilities, not just analytics, but in your process engineering, your formulations, your sensory testing, you name it. We are going to help you be diligent in the details and expect the unexpected. I guess you've heard, I did work at Amyris and Zymergen. Amyris hired me as their first protein engineer. At Zymergen, I was their first director of what we call test operations. So I built out all of Zymergen's initial capabilities for stirred tank fermentations, analytical chemistry, high throughput screening. You can see how we've really harnessed that experience into the design of our platform. We built something that can basically bring all of those teams and the people who engage with them onto the same page. Thrilled to be working with my friend from grad school, Li Kung, who was the director of bioinformatics and software at Gen 9 when it was acquired by Ginkgo. Excited to have Doug Cameron, obviously a luminary in the field, our board, and Martin Otterson, who was with OSIsoft for a decade. He's very excited to basically bring radio to all these people who are used to using the Pi historian in their manufacturing plants, realizing that there's still so much data and so much value being left on the table. Radio is going to help them extract it. So now let's complete that U-turn. We'll sail to the end of this presentation. You've heard other speakers today talk about this same problem. I think we diagnose it a little differently, though. Why is it hard to find the data we need? Because there really aren't alternatives, right? We're just going to have detailed recipes and metadata that simply are not going to exist in any instrument, and pretending otherwise is going to lead you to the wrong solution. Now, of course, there's the machine data as well, and we really want to capture both what I call the human data and machine data. And so there's this second challenge, which is that even when we have some of that data in front of us, it's still very hard to make sense of it. Again, I think the diagnosis here is really important. You make the wrong diagnosis, you end up curing the wrong disease. So, the challenge here is it's called R&D, right? If we aren't changing what we do, we're not being successful at what we do. And software, conventional software, works best when things don't change. So radio has really been designed to enable change. And the way you do it is you make a system that is configurable by the scientists and engineers who are doing the work themselves. As I said, Protein Brewery is able to maintain the system in about 2 hours a week. They were able to build the system without stepping out of the lab. They were able to keep their day jobs. They didn't get sidetracked into becoming, software or data engineers. They remain process engineers. They're just very data capable process engineers. In a nutshell, a lot on the slide, but in a nutshell, what radio is doing is integrating both the human and the machine data. It's doing it with, call it, automatically curated relationships for advanced analytics. No joke, you can literally surf all the relationships in your data using radio. It's like, you can make those shortcuts in Google Drive or SharePoint, but of course you don't because that's work and we have lots of other things to do, and it would be awful. It would be a mess. Imagine like hundreds of shortcuts in every folder on those platforms. In radio, there actually are no folder trees. There is just this dense mesh of automatically curated relationships. You get to surf all these different threads through your data, your human data, your machine data. Radio automatically generates these block flow diagrams to, again, get everybody on the same page. You can share them with each other with automatically generated URLs. And those kind of control charts that I was showing you earlier, you can not just make them in our platform, but you can bookmark them and check them as soon as the latest data is available. Again, we're not going to take away anything that's working for you. You like JMP, you can keep using JMP. Data will go straight from our data warehouse into JMP. You like Excel for dashboards, you can move the data straight from our data warehouse into some dynamic and frankly very powerful Excel-based or Power BI or Power Query-based data analysis and visualization as well. Yeah, so process data fabric, again, scientists and engineers can rapidly prototype and iterate very effective end-to-end tooling. Our web app enables you to really find not just the measurements, but the data files themselves. Our users are telling us that, you know, when they want to go look for a CSV or spreadsheet, they prefer finding it with radio to going to their conventional shared drive. People who've worked with me know how near and dear this is to my heart. Like, everybody should have a control chart, right? Protocols and training, even automation, are simply not sufficient to control the work we do on our path to commercializing new bioproducts. We really need feedback loops. We need 10 times more feedback loops. We need 100 times more feedback loops. There's too much waste in the way we are trying to build this industry, and as a result, too many companies and projects are running out of cash before they have a chance to really deliver on what is absolutely achievable outcomes. I guess one real quick slide, just pointing it out there. In addition to our software and our implementation services, we also offer a workshop. If you are interested in learning how to do in-spec R&D, basically bring that manufacturing mindset of control and continuous improvement in upstream into R&D, make sure that all the work your R&D teams are doing is fit for use and not burning your cash and wasting your time. This is a lot of fun. We get a lot of great reviews. Be happy to discuss it more with you. Last slide. Again, we started with the end in mind. So you've heard this before. Be diligent in the details. Radio really is the missing piece. It's the thing that we didn't know we needed to basically unlock the latent power of our existing team, tools, data infrastructure. And we are going to put all that data at your fingertips so that when unexpected things happen, you can detect them more quickly, more easily. Premium product, please write us, waitlist@radio.com if you want to try. And Biomade teaming opportunity. No joke, we will, in a very non-disruptive way that I believe won't cost your company anything, we will get you 4 times more productive. That's it.
Great. Thanks, Tom. I think we're right at 2 o'clock. So I think go ahead and feel free to reach out to Tom at the contact info here or drop your questions in the chat. I see we have our next speaker on, Liz Chapman from the National Renewable Energy Laboratory. So Liz, if you want to take over the screen here, Liz is going to tell us about integration of software and piloting to help minimize costs for bioprocess scale-up.
So I am from the National Renewable Energy Lab. I've only been there for a little over 3 years. Before that, I worked in oil refining. So I have a much different background than a lot of you who work with the ABPDU. However, I am learning a lot about fermentation, and that's part of what I want to talk about today. So we are revitalizing our integrated biorefinery facility, and we are putting in a lot of new equipment as well as a lot of new capabilities that to update the space. And so as a part of that, we are looking at a few core concepts. We're doing plug and play so that we can move equipment in and out. And that way we can move equipment faster than we can now. Currently, we have 2 160 liter bioreactors, 2 1,500 liter, and 4 9,000 liter. And we can't reconfigure the equipment around those basically at all. It takes us over a year to do so because there's a lot of steel in the ground to get those in place. We are reconfiguring things and moving equipment around so that we will have a more open space. And that way we can also bring in a lot of downstream separation equipment. And we will also have the ability to work with flammable material, which we currently do not have in that facility. We can do it in other places on our campus, but this will be new for this particular facility. One of the other major points is integration. So we have all these great bioreactors. We've got pretreatment reactors, we've got bioreactors, we have paddle mixers. We're also going to purchase some house equipment that will be more on the thermochemical side of things, but that you can integrate to the back end of your biochemical process so that you can do something with it. The other thing we were looking at, it's not funded at this point, but we do have there's space in our minds and space at the facility. We are looking at like some different carbon capture capabilities and doing some different electrochemical research as well that we can integrate into like a fermentation, for instance. Along with all of this, we are also upgrading our analytics system. We are bringing in a lot of online and offline analytic machines, and all of our instrumentation is being upgraded in the facility as well. We want you to be able to do heat material balances without even having to think about it. So that's kind of how we're approaching this. And then with that, we also offer a lot of different modeling capabilities. We not only have CFD, but we can do process modeling. We can do kinetics. We can do a lot of different things. Our staff has a very wide range of capabilities. So we are here to help support whoever needs the support. And we will also offer scaled down options. So if you're coming in and you don't necessarily want us looking at all of your data all the time, and you just want to collect some stuff and move on, and you have engineers on staff who can do it, we can offer that as a solution as well. So one of the main things that we are trying to accomplish with this is full integration so that you can prove your TRL 5 or 6 pilot plant capabilities. So we are on the larger side as far as volume goes. For instance, ABPDU has a lot of bioreactors that are under 300 liters. We're bigger than that. And so we want to be able to take, a commercially available feedstock, process it all the way through and give you a commercially available product at the end. That's the entire point of this revitalization project that we're doing. And so all of that capability will be housed within this one facility. We also are sizing things to produce about a barrel per day, so 42 gallons or so of a sustainable aviation fuel. That is our current goal. And I say that with a bit of a grain of salt. We can do less. Less is always easier, but we have to be able to have the utilities to support the larger scale. We will not be able to do multiple barrels per day. I do want to put that out there. We are not a production facility, but if you do need around 40 to 50 gallons per day of some sort of material produced, we should be able to handle that for you. So, what does that look like on our side? What can we provide you with our house equipment and house utilities? We have bioreactors, we have pretreatment reactors, those are already in place. We have centrifuges, we have a lot of those already in place. We are planning on purchasing fixed bed reactors to do hydrotreating. We are also planning on purchasing distillation columns so that you can do solvent extraction or liquid extraction. All of these equipment that is that is a house equipment piece is able to be re-engineered within a certain realm of operability. So if you want to run something that's a little bit different than what we planned on, we might have to make a few modifications, but we should be able to accomplish that and move on with your project. In addition to those things, we are also supplying a lot of common ordinary utilities, air, high and low pressure steam, nitrogen, cooling water, etc. The biggest thing that we are including here that's new for us is a hydrogen electrolyzer that will be producing green hydrogen on demand. So if you need that for your chemistry, we will have that available to you as a feedstock. Now, how does all of this play into AI, right? Well, we're planning on putting heavily instrumented equipment in that will not only integrate with our LIMS, but with our data historian that we're also putting in so that we can manage the data that you're collecting as you operate. We will have over 30 machines that are available for you to use either offline or online. And that is dedicated only to pilot plant personnel. So if you bring your project to our pilot plant, you have first dibs on what you would like to use. We also have, and this is new to my directorate, so my arm of the lab, our fuel testing capabilities are now much closer to where the pilot plant sits versus where they were before. So we can definitely work with that group and are planning on working with that group regularly to do any fuel testing capabilities that you may or may not. So all of that data, we will also offer comprehensive modeling. So we do a lot of techno-economic analysis and life cycle analysis modeling. That's a lot of NREL's bread and butter. And the point of this entire thing is that, of this entire part of the project, is that we want to be able to take a lot of this high integrity data and plug it into these Aspen process models so that we can give you a model that says, here, this is what you ran today. This is what you did. It matches your yields. It matches your operating parameters. And if something is off, we can help you figure out why it's off. I have a lot of background doing that. That is what I did for years. And we will have a lot of support staff available to help walk people through that if you'd like to take advantage of that capability. We will also have CFD modeling that is available for people if you would like to use it. We currently have research going on with CFD modeling and our bioreactors to see, where the discrepancies are. And we plan on not only doing it within the biochemical equipment that we have, but also in our thermochemical equipment. We do both, and they're pretty good at both. I've worked with our CFD modelers quite a bit, so we can also include that as a capability if you'd like to use it. One last thing that I wanted to bring up here is that we can also vary your power, and we can do power simulation of a green grid if you would like. We do have the Aries project, which is just up the road from us. They are at the other Golden, well, they're not technically Golden, Colorado, but they're the other NREL campus in Colorado. And we can run data from them to your equipment that's operating in the IBRF. And we can change your motor speed based on, if you were doing full solar, we can change electric loads depending on what kind of grid you'd like to see. So that is another capability if you're looking at things like a more modular system that you wanted to implement in smaller places that's more off the grid. So the largest thing that we're looking at right now when it comes to AI in this particular revitalization project, excuse me, is that we are looking to use a data historian that feeds our process operating data directly into a process simulation. So those process simulations can run sensitivity cases based on the operating data that we're seeing. that not only saves on your operating costs and piloting, because it can predict the results of what may or may not happen, but we can also then, as a process engineer, this is my job, right? I look at your operating envelope that you've established and said, we have major success in this operating realm. I can tell you where to push it or where to pull it, to test those limits. And that way you have the most robust case of data that you can have, and you know where your failure points are and where you can't exceed things, or where you maybe could push it a little bit more as you continue to scale up. And that will help you become more and more successful, because as you scale up, you need to know where your failure points are. If you don't know where those are, then I think that you have a lot of risks associated with your scale up. The other thing that we are doing is we're taking all of that a step further and looking at projected yields, and I would like to audit. So I want to go then, let's say we set up a case, this is your base case, and we know that this operating envelope works. We stretch your operating envelope, and your process simulation says you should expect X, Y, and Z on your yields. Well, I want to go run that. And I want to make sure that we are actually getting what the process simulation is telling you you're going to get. That way your data is valuable and you have high integrity data at that point, and you have a lot of confidence in what you're seeing. So then you can take your process simulation and just move that instead of moving your operation. And it's a lot less costly to you as a customer. So with that, we have a lot of scale up support that you have access to at NRail. We have a lot of experience, be it process engineers, control engineers. There's a lot of us who have operated commercially. There's also a lot of us who have operated large pilot scale things, and know what troubleshooting needs to look like so that we can help you move through different hiccups that you're going to encounter as you scale things up. We are also planning on having some technical service support from the engineers that are already on staff, if you'd like access to those people. And they can use the AI modeling tools and the information that we're collecting to help give you a roadmap on how to operate and inform your decisions as you start moving forward with your scale up. There was one other aspect of AI that we were also looking at, and that is predictive maintenance. So there's a lot of software that's already out there. And I think how we're going to approach this right now is we're just going to test a few of these for a year or two. Predictive maintenance isn't necessarily the best fit for piloting, and we completely understand that. It's meant for commercial units that are operating for thousands and thousands and thousands of hours. But we were going to do a study to see if the software might be effective for scale up, to figure out if there's any predictive maintenance that we can say, hey, you might have problems with this compressor skid. Think about, doing a duplicate or a parallel skid if you're building a commercial unit, kind of a thing. A lot of that, we'll see. That's something that we're looking at. I'm not 100% sure what the outcome is going to be yet on this, but it is on our radar. And it would be something that you would have access to if we move forward with this study. So generally, the IBRF is about 6,000 square feet. That includes both our South High Bay and the North High Bay. We are currently in the process of designing it right now, and that design process will probably take us another 9 months to a year. But the construction is scheduled to be completed sometime in mid to late 2026. Excuse me. So we do anticipate being able to bring in new equipment in late 2026, early 2027, so that you can start running in our new facility. We also, just to throw this out there as a separate thing, we have another facility that's being built in, hang on, this space right here on our campus. It's another large lab building. We will have BSL2 capabilities there and a very large pilot plant there as well. That isn't tied to my project directly, but I am very well aware of what's being put over there. So that one won't be ready until 2027, maybe a little bit later, just because it's a very large building. And right now it's just a hole. So we have some work to do, but it will be available for people to use in 2027 or 2028. We do have a large team that is working on this. These are the people who are involved in equipment only. So I think some of you may know Dan Schell. He's our operations lead at the IBRF. Excuse me. Some of you may also know Jesse. I think he's been on today's call. He's on the Agile Foundry, excuse me. And he's our business development lead for our project. Excuse me. And then we have several other researchers that are also helping us. But if there are any questions, we'd be happy to answer them. And we can be reached at pilot.plant@nrail.gov. That's a general email that we all check. We all have access to it. So someone will get back to you if you email us there. And thank you all for your time today. And that was much faster than I thought it was going to be.
Oh, thanks, Liz. Well, plenty of time for questions and discussion here. So I guess quick question for me would be, or could you kind of walk us through the process of partnering with NREL on these projects, and how are these structured? And are you open to sort of anything within biomanufacturing? Are there specific types of projects that you're especially keen on getting going?
We're really keen on any scale-up projects. So if you're looking, like for instance, you're working with the ABPDU on something that's 100 liters or somewhere in that realm, we can help you take that from the ABPDU and then put it into like a 1,500 liter fermentation or larger. That's kind of what our bread and butter is. We also do technical service agreements where we can run a larger fermentation or for you and get a certain amount of product. And then we also have CRADAs, which are our research and development agreements. And those are like the main ways that you can work with us. The TSA, the technical service agreement, is the faster way. But the CRADAs are really good for larger long-term projects. So we have several different things, and you can always contact Jesse or this pilot plant at nrel.gov to get more information. But we have a lot of different mechanisms to work with us.
Yeah, thanks. And so you mentioned your LIMS system, and we've been talking about software a lot today. I'm wondering, could you go into a little more depth on kind of what sort of system you have on site? Is it in-house, or is it a third party? And is it integrated across all of your platforms?
We have, I'm fairly certain it is an in-house LIMS system. It is not integrated across all of our platforms at this point. I think that's happening within my directorate fairly quickly compared to the rest of the lab, because we're getting more and more spread out as far as pilot plants go. So we need to be able to access the data. That being said, this data historian is a new thing for us on NREL's campus. I've used them a lot in my previous life, and that's why I'm pushing us to move that direction. But the LIMS system, we are integrating it into the data historian. So we'll have to make tags for all your samples and everything, whether it's online or offline, but that way you can pull it in real time. Sorry. And it should help with we don't want to duplicate efforts, right? So we're trying to eliminate that with this LIMS system as well.
There's, like, we have a question in the chat here as well. So the question is, does the pilot plant support consulting partnerships to help startups launch lab scale experiments with the goal of reaching your minimum scale of operation? So would you support partnerships that aren't ready for scale up yet, that could create a pipeline towards scale up?
Absolutely. We have 2-liter bioreactors all the way up through, like I said, 9,000 liter. And we do a lot of lab work. So we are able to help you with that. We helped, it wasn't bio-based, but, or sorry, it was not biochemically based, but we did help a startup on lab scale get all the way through. And now they're working with the Department of Energy on a scale up FOA, I believe. So they've moved like we've gone from bench scale all the way through piloting, and now they're working on even larger things. But that's what our team is really good at, is helping you get from lab scale all the way up through what our piloting capabilities are and beyond.
I know many groups, many groups are looking at private CMO for similar types of work. This is a question we get asked a fair bit as well. So how would you think about differentiating what NREL offers in this space versus what somebody might get from a contract manufacturer elsewhere?
We are not a, what's the word I'm looking for? We are here to help you with your technology and your scale up. We are not here to help you produce gallons of fuel or gallons of product. That is not what we want to help you with. We're here to develop your technology and help you get your IP out there and commercialized. And so we are not, we're not going to be one that's quick to turn things over and say, you know, here's your final product, and then you leave and we never talk to you again. We want to be a partner that helps you move your intellectual property all the way up and get out and commercialized. So that's a huge thing. We have a lot of technical support that does that with our modeling, our TEA and LCA modeling. We can help you with all of that. A lot of those other commercial plants can't necessarily do that. So that's where our expertise lies, is in that support part of it. I think there was another question in the chat.
Yeah, there's one more. So the question is this pilot plan at NREL part of the Agile Biofoundry activities, or is it separate? Which,
hey, Jesse, are you on? I don't know if you can hear me.
Yeah, hi, I'm on. Can you hear me okay?
Yeah, do you want to answer this one?
Yeah, sure. The delineations between different organizations within the Department of Energy labs, I think, can be a little daunting for folks who are not part of those labs to really appreciate. And I'm not sure that they're terribly important to understand as an individual entity coming in, unless you have, you know, a particular funding arrangement or intellectual property management plan with a specific lab. So if whoever asked the question wants to talk about it more in depth, I'm happy to answer the question more appropriate to what underlies the question. But all that said, the IBRF does execute on projects that are supported by the Agile Biofoundry. Some of our bioprocessing development and optimization work has been done in that facility. The facility itself is not directly funded by the Agile Biofoundry. So I'll leave it there, but if I did not answer your question in spirit, please just reach out to me and I'll do my best to understand the question better.
Thank you, Jesse.
Yeah, I think that's helpful. I think from the outside, the programs look very, it's very hard to tell the difference. I think functionally for external collaborators, it doesn't matter too much in that IBRF and ABPDU can all be accessed as scale-up facilities for ABF projects. So
yes,
functionally works well.
Right. And the other part of that is that, like, all of our funding for the IBRF is given to us through the Bioenergy Technology Office. They support our day-to-day operations and our maintenance. So it is not necessarily tied to any sort of consortium, because we also work with like the CO2 consortium, and I know the ABPDU does as well. So there's several different Department of Energy groups that we work with, but we're not supported by one necessarily.
So our next speaker is Ishan Shrivastava from Berkeley Lab. Ishan is going to talk about bioreactor optimization using computational fluid dynamics and multi-phase flow modeling. Ishan, go ahead and take it away.
So yeah, thanks, Eric, and thank you, the organizers, for inviting me to present some of our recent work in bioreactor modeling in this forum. Yeah, as I was saying, so I'm Ishan Srivastav. I'm a research scientist in the Applied Mathematics Department at LBNL. Our group and our department are primarily funded by the Office of Science of US Department of Energy. And my group is typically, our research is in developing computational methods for solving large-scale scientific problems that are multi-scale and multi-physics in nature, and which are typically described by partial differential equations. So my background is not in biomanufacturing or bioreactors, but it's been a couple of years since we realized a lot of our models are applicable here. So I'll show you an example, and I'll finish with what our planned roadmap is in this area. So yeah, I don't need to obviously motivate this audience about what are the opportunities in bioreactor scale up. This is a slide that I picked up from a Boston Consulting Group report from 2024. And the idea is that biomanufacturing of mass products, such as chemicals and food, holds tremendous promise for greenhouse gas reduction, which is often the mandate for DOE national labs. However, the challenge is that there's currently a 30 to 50% price premium over traditional manufacturing. So in order to make these things more cost competitive, it is the case that the manufacturing needs to be scaled up, and it needs to be scaled up by an order of magnitude. However, as it was alluded by several speakers before me, there are significant challenges associated with scale up. The two technical challenges that are relevant to what I'm talking about are the case that as we scale up any chemical reaction process, and certainly the case for bioreaction and bioreactors, we see increased heterogeneity. So a typical pipeline of development of a bioprocess starts at a lab scale, which is around a liter of a bioreactor, which is more of a proof of a concept type of a scale where strain engineering is being developed for a certain microorganism, and typically things are very well mixed in the bioreactor here, and we don't have to worry too much about heterogeneity. But as we move on to pilot scale, which is where process development happens at approximately 100 liters, and eventually to production scale, which is the case when commercial production happens, what we see is that there is increased heterogeneity both in the fluid dynamics of the problem and in the metabolic distribution in the system. So this particular figure that I picked up from one of these papers elucidates this point very, very nicely, where what happens is, because of large scale heterogeneity in let's say the level of oxygen or the level of glucose, it could be the case that there are certain cells that are substrate restricted, there are certain cells that have overflow of substrates, and then there are perhaps few cells that are in a normal environment. Additionally, there is also heterogeneity in the external environment. So the fluid shear stresses would be very, very dramatically distributed. There would be regions of very high shear, presumably near the impeller, and high shear stress can kill the cells, right? There could also be very strong concentration gradients, both of the substrate and the oxygen. And this increased heterogeneity is often a challenge to essentially optimize for yield and optimize for cell health over a long period of time, over an entire campaign of a bioreactor process. The other challenge in bioreactor scale-up is that the rheology of the entire system is pretty complex, and it's transient. So this is a bunch of images of a bioreactor in operation that I got from this reference, where it shows that even visibly one can see things changing dramatically from the start of the process to the end of the process. And what these researchers did was they monitored the amount of torque that is being used to move the impeller at a certain RPM as a function of time, and even can see that things are highly intermittent. There's a significant rheological change with time. Things get more and more viscous. in an often unpredictable and unoptimized manner. And this can lead to high energy consumption during the bioreactor operation. And it is often the case that that aeration and impeller motion can is a big percentage of the total cost of a bioreactor operation. And furthermore, by as the rheology of the system changes, there are significant issues that happen with the mixing in very large reactors. So clearly there are some very significant technical challenges associated with scale up in bioreactors, and these two in particular are something that our group has a bunch of computational methods and computational frameworks that are well posed to tackle them. So what is really needed from a computational perspective to advance bioreactor scale up is a modeling framework that couples biokinetics to a computational fluid dynamics model. And the previous speaker did allude a bit towards CFD modeling. And in this presentation, I will show you some of the features that we can get from these CFD models. And an ideal CFD model, what would happen is that there would be a way for us to track particles, which is cells or collection of cells. We resolve for the velocity field throughout the entire bioreactor. This would be coupled to the concentration field of substrate, glucose, oxygen, carbon dioxide, etc., or of different metabolites, whatever is of interest, right? And these would basically be talking with the biokinetic model. The biokinetic model would be the one where the actual biochemical reactions would be happening at the cell level. So for instance, there would be cells that would be carrying the information of their local metabolites, the local concentration of glucose, oxygen, carbon dioxide, etc., in addition to some fluid dynamic variables such as velocity field and shear stresses. And it is in such a model, what one would be able to get is a differentiation of certain cells in certain regions that are starving, right? They don't have enough substrate. There are certain regions or certain cells in certain regions that have an excess of substrates. And finally, we can look at which cells are resource limited and which are not. So a computational framework of this kind will essentially be able to provide very high fidelity spatio-temporal information of what's actually going on inside a bioreactor. Because as we know, looking inside a bioreactor is extremely challenging, and there's a lot of ongoing developments on incorporating advanced biosensors. But regardless, I think there's a place for physics-based modeling of these bioreactors that can really provide information on what's going on inside a bioreactor. So with that motivation, let me show you what our group has done historically over, I would say, the last 20 years. So our group at Lawrence Berkeley National Lab in the Applied Mathematics Department is home to a software suite called AMRX, which is Adaptive Mesh Refinement at Exascale. And the idea behind this software suite is it solves partial differential equations, mostly related to fluids, but we have expanded into other areas, including astrophysics, cosmology, electromagnetics. But for this purpose, we can restrict ourselves to reactive fluid dynamics. And the power of our software suite is that it is adaptive mesh refinement in nature, which basically means that we can look at certain regions of a simulation very closely by adaptively refining. So if you look at the second GIF image here, you can see that this is a case of a complex fluid. So this is a case. This is a fluid that's undergoing shear from the top. And its rheology is very complex, not unlike the one that I showed you in the bioreactor. And what is happening is that there are certain regions that are becoming more and more featured, where there's some bubble is breaking or a bubble is coalescing, right? And our software essentially provides the capability to zoom into those regions without refining the entire simulation domain, which would be extremely expensive. We have developed methods for reactive fluid dynamics over the last 20 years. So on the left, you can see an example of a burner. So this is carbon dioxide. This is hydrogen coming out of a nozzle, which is then getting combusted with oxygen. And it's basically a low swirl burner. And it's an extremely detailed simulation. It was actually the most refined simulation of its time when it was published. So these are the kind of simulation methods that our group has developed in the past and has an expertise in. We also have the capability of modeling the motion of solid particles in a fluid. So on the right, you see a chemical loop reactor, which basically moves around particles in a fluid and it provides for a way to exchange carbon dioxide and to store carbon dioxide. So this is called a chemical loop reactor, and we have been able to model this at the scale of an actual production, which basically contains up to 10 billion particles. Our code is performant on leadership class exascale supercomputers with GPU acceleration. Our code has been tested on pretty much all of Department of Energy National Labs exascale supercomputers. It provides support for multi-phase fluid flows with reactions or with surface tension between the phases, which would be the case in separation, filtration, or in just simply modeling the bubble dynamics, right? And we also provide support, as I said, for solid particles or bubbles in fluid flows, along with moving boundaries, which would be needed for modeling the impeller. So in the next slide, I'll show you an example where our group has been collaborating with a company, Capra Biosciences. It's a startup based out of Virginia. And we have basically been modeling their bioreactor operation using our code MFix-Exa, which is an AMRX-based software for multi-phase flow. So I won't go into the details of what I'm showing you in a simulation here. These are highly detailed simulations. But basically what we are solving for is bubbles and feedstock coming in from the bottom that's being force pumped from the bottom in a fluid, and then there's a bunch of biochemical reactions happening, and then things are leaving at the top from the pressure outlet. So this is a level of detail that we can model using these simulations. Of course, I won't go into the details of the equations, but we are basically solving for fluid and the motion of feedstock particles and the motion of bubbles in this simulation framework. This code is highly performant. We were able to scale it up such that 60 seconds of simulation time or 60 seconds of bioreaction operation time was of this fidelity could be simulated within 2 hours of computer time. In this case, we used about 80 NVIDIA A100 GPUs on Oak Ridge National Lab supercomputers. This was Summit supercomputer, but we believe that some of the more latest ones, such as Frontier and Aurora, we can get even better with this scaling. So as you can see here, we were able to track local fluid velocity, local velocity of the gas bubbles, local velocity of the feedstock, and even things like what is the shear stresses experienced by feedstock particles and the bubbles, because our industry partner was interested in looking at high shear zones, because that's where they were expecting cells were probably dying. One of the learnings from this simulation for our industrial partner was that we identified certain dead zones in their bioreactor where there was presumably not much mixing happening and things were not moving around, which was essentially affecting their yield. And based on our simulations, they were able to optimize their bioreactor design, such as location of inlet nozzles, etc., so as to get better mixing in the bioreactor. We simulate our processes on 2 modes of operation. So what I showed you was a coarse grained simulation, but we can do something that is more fine grained. So for example, on the top, what you see is something called as a discrete element method, where we basically model each gas bubble or each particle, and we track its motion in time and in space as it moves around and it's advected by the fluid. This is the highest fidelity mode of operation. It has the least fitting parameters, and it is great for calibration. So with our industry partners, we basically first ran a discrete element method simulation of a bioreactor. This is highly expensive, so because we are tracking each individual particle, but this gave us a good notion of what's happening locally in the dynamics of the bioreactor, and we are in the process of comparing visually, at least, the dynamics predicted by our simulation with their small pilot scale bioreactors at their facility. The learning from this highly resolved DEM simulation then eventually feeds into a more coarse grained method, which we call as particle in cell method, which is what I showed you in the previous slide. So here we basically model a collection of particles or bubbles instead of each individual one. So it's obviously lower fidelity, but this is the one that can be scaled up to very large scale, even commercial scale bioreactors. I won't go into the details of the size of the bioreactors that we simulated, but yeah, particle and cell method can essentially enable us to scale up to 10,000 or even 100,000 liter bioreactors quite straightforwardly. But yes, it is of lower fidelity and it requires calibration from a high fidelity model. But yeah, we have the workflow set up to be able to do something like that. And finally, what I showed you is a very detailed simulation, but quite often what we learned from our industry partners was that they often don't really want to see the motion of each individual gas bubble or all solid particles, but they would rather see them as continuum fields. So for that, we are working with certain collaborators to use our other software, which is incompressible fluid flow software called InkFlow, where everything is basically be treated as a continuum scale, and each cells are tracked as individual tracer particles. So the two cells basically don't interact with each other. This is the least expensive option that we have, and we can interface it with bioreactions, etc. I will move quickly. Some of the ongoing developments that we have right now is coupling our CFD models with a kinetic model, where now we can have biochemical reactions also in our modeling framework. And lastly, our eventual goal is to basically develop a data-driven coupled CFD metabolic modeling software with optimization capabilities. So we are working with folks in the biosciences group to essentially use some of their ML developments in our bioreactor simulation. I will end with this slide. This is the team that has been involved in this project over the last 8 months, from Berkeley Lab, Mathematics Department, NETL, and Biosciences Group at LBNL. And I would like to acknowledge Capra Biosciences, the industry partner that we have worked with. Thank you.
Thanks, Sushan. This is really powerful stuff, and hopefully it finds a lot of relevance with the folks on the call here. So please drop any questions for Ishan into the chat. And also thanks to everyone for sticking around until our last talk here. And thanks to Michael Clear, our last speaker, for closing us out on day one. So Michael's going to speak about a pre-competitive knowledge base at Schmidt Sciences. So Michael, I'll hand it off to you. Feel free to take us home.
So first off, thank you to everyone for sticking around. Thank you to Deepti and event organizers for giving me the opportunity to talk about this project that was funded by Schmidt Sciences. So my name is Michael Clear. I'm a science associate at Schmidt Sciences, which is a philanthropic organization founded by Eric and Wendy Schmidt. So my goal is I want to talk about this pre-competitive knowledge base. It was a program that we funded, but I also want to give you a little bit of background on Schmidt Sciences because we are a relatively new organization. So we officially launched in February of this year, but many of our efforts actually predate February of 2024, as we were originally part of Schmidt Futures, which is a philanthropic initiative, also part of the Eric and Wendy Schmidt philanthropic entities. At Schmidt Sciences, we use a variety of mechanisms to support early stage, high risk, high reward research that ranges in scope from basic to fundamental science questions and all the way up to major problems that face the planet. And so the work that we do is broken up into 5 different focus areas. We have an AI and advanced computing focus area. We also do astrophysics and space, biosciences, which is the team that I'm a part of, climate, and then also science systems. Science systems may not seem quite so self-explanatory. That group mainly focuses on a lot of fellowship programs, but also interdisciplinary projects. One of those programs that fits within interdisciplinary would be creating professionalized open source software for scientific teams. So as I mentioned earlier, many of our programs predate Schmidt Sciences. And so actually the biosciences group was kind of born out of a task force effort that was created by Andrea Hodgson and Mary Maxson. As part of this effort, they convened people that were former regulators, academic researchers from biomanufacturing and biotechnology, as well as those that were involved heavily in industrial biomanufacturing. The goal here was to try to create a U.S. bioeconomy strategy. So, how can you focus on what are the main bottlenecks to creating a vibrant circular bioeconomy? And so just kind of a high-level overview of the findings of that report is that there are 5 main pillars that needed to be focused on for creating this bioeconomy. The first being focusing on expanding R&D efforts as it related to translating findings from the lab for public and economic benefit. This is quite broad. Examples of this could be something like genetic and enzymatic pathway modeling, up to whole cell modeling all the way to cellular community modeling, specifically as this relates to designing software that allows better bioprocess models. This also could include research such as creating waste biomass or processes to convert waste biomass feedstocks to useful bioproducts, as well as engineering challenges, thinking about developing purpose-built bioreactors or sensors for biomanufacturing. So the second pillar is focused on fostering a national ecosystem of innovation and commercialization. This would include data that are relevant to, or activities that are relevant, such as the pre-competitive knowledge base that I'm going to talk about. So that's thinking about creating a data and computational infrastructure to support emerging and small businesses and product development. Third, we have building a national infrastructure for bioproduction and scale capacity. That's thinking about places such as the ABPDU. We also have developing a diverse workforce to power the current and future bioeconomy. And then finally, enabling policy that incentivizes and supports circular bioeconomy. So this is, I promise, my last slide about Schmidt Sciences before getting more into the pre-competitive knowledge base. But I do want to talk a little bit about some of the reports that have come out, and one of the mechanisms that we often use when thinking about what programs that we want to scope, and some of our internal strategies. So often it's really important for us to have workshops and convenings when we're thinking about moving into a particular scientific area. And our goal of those convenings are twofold. One, we do want it to inform our strategy and how we move forward with the program, but we do also want to have, an artifact, a leave behind that's hopefully helpful for the scientific community. And so I do want to highlight a few of the reports that have come out over the years. The first one being a Feedstocks of the Future for a Circular Bioeconomy report that was put out by the Foundation for Food and Agriculture Research. This was put out in July of 2023. In addition to the report, this informed a program that we kicked off this year entitled the Virtual Institute on Feedstocks of the Future. Please feel free to follow up with me if you have any questions about these afterwards. In May of 2024, there was a project led up by Imperial College and EBRC focused on global metrics and standards for engineering biology. And then moving forward, we are working on a workshop report that's a result of a workshop focused on purpose-built sensors for biomanufacturing. So you can look forward to that in 2025. And as I mentioned, my focus here is going to be talking about establishing a pre-competitive knowledge base. And this was work that was done and led up by the Society for Industrial Microbiology and Biotechnology. So I know this is coming at the end of a long day focused on data. I don't feel like I need to belabor the importance of data sharing mechanisms, but I do want to outline that, not just in our task force report was this a really important bottleneck to focus on, but many of the reports that have come out of federal government, the private industry, have outlined action plans for having homogenized data that can be utilized to advance the bioeconomy. I did want to highlight this interagency workshop report that was focused on the data for the bioeconomy. This was one of the products that came out of Biden's executive order for biomanufacturing and biotechnology. Just one, they highlighted that it was really important to have shared ontologies and sustained data resources that could increase the speed and scale of biomanufacturing. And they highlighted some specific data needs, including integration, linkage, quality, interoperability and reusability, data generation, monitoring assessment. So these types of ideas are what informed why a pre-competitive knowledge base would be important, but also how would go about developing a pre-competitive knowledge base. So when it came to who actually led up this project, Nigel Mounsey, who's formerly at SIMBI, who's now a director of the Joint Genome Institute, was the chair of this effort. Also had some amazing co-chairs, including Deepti Tanjore here at ABPDU, Chris Stowers at DSM Firmenich, and then Haley Cox, who's executive director over at SIMBI. And so in terms of the report development, this was there were 3 workshops that were convened, 2 in 2023 and 1 in 2024. And the idea of these sessions were to explore the potential of a platform to bridge the gaps between foundational research and commercialization in order to enable faster, cost-effective development of these bio-based products. So the idea would be that the pre-competitive knowledge bases, or PCK's core value, would lie in its ability to reduce the development time from 10 years to 5 years, and development costs from what typically are $100 million down to the order of $50 million. And it would achieve this by structuring and sharing pre-competitive and post-competitive data, which is currently either inaccessible or in fragmented forms. So in terms of what is pre-competitive data, these are things such as omics data sets, bioprocess models, and regulatory insights that could drive innovation by reducing redundancy and revealing new opportunities. And then post-competitive data is something like insights from past commercial products, so you can help avoid the pitfalls and provide more proven pathways to commercial success. So in order to succeed, the PCK would need to address a number of challenges. We need to think about data quality and usability. So this would be ensuring data is trustworthy, standardized, and structured for interoperability. You need to incentivize contributions from different stakeholders. So this would be encouraging public and private stakeholders to share their valuable data. It would need to be accessible, but also secure. So you need to balance open access with data confidentiality and intellectual property protections. And there needs to be governance and sustainability. So there needs to be a defined management, funding, and long-term operation structure. So the goal, again, would be to try to have PCK exist so you could foster collaboration and streamline the commercialization process in order to help the bioeconomy. If you want to see more information about the SIMBI report, you can go to simbihq.org/PCK. There's some additional information on that website, as well as a link to that report. So I imagine a number of you are quite familiar with biomanufacturing readiness levels. When it comes to PCK, the goal, again, is to bridge early stage research, so that's thinking scales 1 to 3, with late stage research, so that's 8 to 10. And so PCK is meant to focus on readiness levels 3 through 8. And so it is important when developing the PCK that it would need to be cognizant of the specific needs for each bioreadiness level. When starting to think more tangibly about what a use case could look like, one example would be sharing media recipes for common biomanufacturing hosts, such as E. coli, Saccharomyces cerevisiae. This could help early stage startups that typically have to invest heavily in media development that allows them to generate high density cell growth at scale, whereas many of these late stage companies already have this information. So now thinking a little more about how would a PCK function in the data landscape, what type of data you'd want to focus on. The PCK, as envisioned by the workshop and the report, was meant to be both a data repository and data facilitator platform. So as a data repository, the PCK would house technical data, such as fermentation media, operational conditions, microbial physiology, and product recovery methods. This data would need to be structured and needs to have appropriate metadata and standards to ensure searchability, retrieval, curation, and integration, and would need to adhere to fair data principles. And then as a data facilitator platform, the PCK aims to make data easily accessible and would need to be user-friendly using tools such as natural language query capabilities. That way you could cater to a broad user base from technical disciplines like biology and engineering to logistics, policy, and commercialization experts. And so in reference to some of the data types you see on the right-hand side, to ensure a strong foundation, the PCK's minimal viable product would prioritize technical data for fermentation and bioprocessing scale-up, and would also include process data and shared details for processing methods to enable interoperability. And the platform would support advanced tools such as kinetic fermentation or mixing models, while future phases would expand to incorporate additional data types and user needs. And so to try to bridge this gap between early stage and late stage, the PCK does need to, again, have these two core functions. So it needs to both ingest data from contributors, but also provide its user structured data in a structured, accessible, and secure way. So to ensure maximum usability, standards need to be critical. So in the development of a PCK, existing frameworks such as NIST, bioeconomy lexicon would need to be leveraged, but also new standards need to be developed. And this would need to be a working group. So this includes guidelines for data submission, quality, metadata, and statistical consistency. And to promote adoption, the PCK would need to feature user-friendly interfaces, again, to make sure that you're using user-centered design methods to adapt to the evolving needs of contributors and users. And then lastly, it is important that the PCK balance data sharing and confidentiality. So this would be, you need to implement flexible access models, IP protection measures, and disclaimers to ensure stakeholders can contribute securely and responsibly. And so really the big, to kind of summarize the primary findings of the PCK report is there are a number of recommendations and conclusions. So you have the funding part is essential. So initial resources should include scoping and stakeholder interviews to refine the vision and the minimal viable product before development begins. Second, the PCK can either integrate with existing platforms or be independently funded with partnerships formed when appropriate. Third, a user-centered design approach is vital. This ensures the platform reflects diverse needs, including input for educators and international users. And then in order to facilitate usability, we recommend developing a standardized lexicon for terminology and structuring data with metadata and provenance information to enhance the interoperability and reuse. The platform does need to be flexible to accommodate evolving technologies, data types, and knowledge sharing models like AI. And then in order to encourage participation, we propose incentives for both data contributors and users, as well as straightforward data access with clear terms of use. And then this part is really critical. Early successes in creating case studies that demonstrate the tangible benefits of a PCK will be really critical for growing the platform and building trust. And then lastly, a dedicated governing body must oversee the PCK's design, management, and operations, enforcing a code of conduct to ensure accountability and maintain standards. So what I want to leave you with is still some of the identified challenges, because again, this was the result of just 3 workshops, and there's still some major challenges that need to be overcome. So first off, funding. So the PCK concept requires rapid development in terms of timeline in order to be relevant. So it's thinking of something that needs to be developed within 4 years. The estimated costs are between $10 and $20 million a year for development and $10 million a year for sustaining growth post-development. And this would require 20 to 40 people for system maintenance and growth. Some of the unintended consequences of creating something like a PCK are potential risks that include biosecurity and issues with access and benefit sharing. The PCK must be indemnified against liabilities arising from the data use, and there need to be safeguards and protocols to be developed with input from biosecurity experts. Finally, the most critical component that came up time and time again in the workshops is the importance for buy-in and trust. So the PCK's value is going to depend on the data quality, usefulness, and really those case studies that show the value of something like a PCK. And so with that, I will close out. Here's my contact information. Again, please reach out if you have any questions about PCK or Schmidt Science's other programs.
Thanks, Michael. Looks like we have one question in the chat, so we can do a quick question here. And they're just wondering where they can get more information about the Sensors for Biomanufacturing initiative.
The best way would be to email me. I head up that program as well. So please send me an email this here.
Thanks, Michael. And thanks to everyone for sticking with us till the close. We'll be back at 7 a.m. Pacific time tomorrow morning, which should suit some of our European audience a bit better.
Thank you all speakers for today. Looking forward to see you all tomorrow.